is the capacity to do that work. Because a different thing to say, I want to do something, and another thing to say that I can do it, but it's more important whether I will be able to do it. So I will delve a little bit more into each one of these aspects in a way that would drive the message home. So before we get to that point, let's look at the different funding mechanisms. If you're coming in and you want to do this work, you've already been able to have a very clear idea about what you want. And so there are different funding mechanisms. So there are others that are the kind of research that you want to do a proposal. There's an aspect of training. The grant, it depends on which type of grant are you applying for. So we have training grants, we have research grants, we have intramural grants, we have R&D, career development grant, and so on. So again, that's another question that you have to answer on what you want to do. So, so the first thing that then you look at is that you look at the funding mechanism and you look at the organization that is funding. Should you decide with the path that you want to take? And the most critical thing is that you really have to go on and have a very good understanding of the funding organization. Look at their corporate strategy, their mission, vision, values, and try to see whether they fit in line with the application that you're putting in. Because we cannot apply for a grant in engineering to a canton institution that only supports biology, isn't it? So you come down to that level. And then the next thing that you look at is your eligibility. Are you eligible for that particular criteria? What are the eligibility criteria that has been put in? So this usually comes out very clearly in the call. And most of the grants are usually eliminated very easily, or you fail to get funded if you don't read the call very clearly. Because the call clearly shows which are the candidates that are eligible and which category, which aspect of research that is going to be funded. So if you don't comply with this call requirement, then in most cases, your application will not even be reviewed. And even if it's reviewed, there'll be a delay in processing. I think the other most important thing is that at what stage of application are you? We have the early stage career grants. These are usually targeting graduate school, postdoctoral fellows, and pre-basic. And then you also have the early stage funding. And then with the middle career or whether you're a senior. So this is actually one place where I think in the universities and research institutions in Africa, we have actually failed our own people. Because in the sense that when someone graduates from undergraduate, graduates from the university with a PhD, that person is usually considered an early career researcher. And this usually runs into the first 3 years of graduation. And some institutions push that up to around 7 to 10 years after graduation. After that, that person is considered a senior or an established researcher. And this has very clear implications. So this has very major implications on whether the person will be funded or not. The early stage investigators are usually, applications are usually considered with a lot of exceptions. For example, an early stage investigators are not required to have any preliminary data for them to be funded. And they're usually considered more in terms of capacity building for the institutions and for the research community. So they don't get a lot of challenges, and their reviews of the applications don't go through a lot of rigor in the review. But for established researchers like myself and many of us here in this room, our applications go through a lot of rigor in the review, and the probability of success becomes clearly diminished. So one of the things that many institutions also look at is that they look at aspects of whether within that particular career, what is the focus? Are you looking at developing an independent capacity? They also look at whether that is going to develop some local expertise in research and training. and so on and so forth and so forth. So you have to clearly stage yourself on where you are. And many grant organizations have different grants for different stages. That's what you're saying, to put in a nutshell. So in most cases, if you're not sure, every grant call at every application always has someone called a program officer. So if you're not sure of the call that you have and where you want to submit it and whether it will be accepted, always call the grants officer. That's why they are there. And every application comes with that bit where you are given a room to call a grants officer and seek clarifications. That's where you can call and say, I want to submit this application, and you only need to do only a small abstract of what you want, and they will always advise you and save you a lot of trouble and time. So the other aspect that comes in is whether you have sufficient time to do this application. Grant cycles normally takes about a year. A good grant would almost take a year to go through the entire cycle from submissions to funding. And most of the grant applications, wherever you need them, in my opinion, a good grant application needs a minimum of at least 6 months of preparation from the point you start preparing to the point of submission. The most interesting part of it is that if you're an experienced researcher, most of the granting bodies will require that you must present some preliminary data that support what you want to do. If you don't do that, then you won't be funded. Preliminary data usually means that you have to go back to the lab or go to the field and collect some data, analyze, present, and publish. And the other thing that also comes in that is very important is the aspect of getting supporting data. And most of the supporting information that's required is usually by way of letters of support that you need from different organizations that are working with you or that will be supporting your application. And most of them are usually external to your organization, and they take time to provide those letters because all the letters of support must be provided by the CEOs of respective collaborating institutions and not anybody else. And each one of them have their own timelines. And also there are also issues to do with putting together your collaboration, getting the letters of commitment and so on. So wherever a call comes out, before you even think about the science itself and putting together the team and cobbling together the application. So the first thing you ask yourself whenever a call comes in and you read it, and you ask yourself, do I really have sufficient time to submit this application or should I let it pass? And then at the same time, some institutions also have some internal deadlines. Like for example, you might find that if there's a deadline for submission of an application, say by the end of June, if there's a time to submit an application by the end of June, you might find that the University of Eldoret might have the University of Eldoret might have its own internal deadlines, isn't it? Saying that because we have a deadline of 17th of June, we must make sure that by 17th of May, we have everything in place. Because I have seen in most cases very disappointments at the point of submission when you submit on the very bit of the deadline, and then you find that there's some very small document that you don't have that should also be uploaded and is not available. Or whether nowadays most of the submissions are electronic and you find that the electronic challenges for submission, or do you just have a blackout, or you find that there's no internet on the very day that you're supposed to do the submission. So institutions tend to have their own internal deadlines, where they say that if there's a deadline for submission, as we must upload and submit our hours at least a week or 2 weeks before the actual deadline to give us room to do any corrections that might come in. So that comes in as well. So once you have this, the other thing that you need, once you have that out of the way, then you ask yourself a few questions. What processes will this application take? In the sense that, what are the deadlines? Because usually every grant submission application begins with the submission. Then they check the eligibility criteria, the funding recommendation, and then the board approval, then you get a feedback. The point I want to put across here very clearly is that most of us only think that the people who decide who gets funded is usually the reviewers. That's the feeling that we have, which is actually a misconception, in the sense that once the reviewers submit their reviews, they write back to the funding body. The funding body actually provides recommendation on who should not be funded, but the people who make the final decisions is usually the board members of that organization. Like say, for example, if University of Eldoret was a funding organization, the people who would actually decide who gets funded or not would be the university council and not the university management. The university management can only recommend, but the final decisions come to the council. And this is going to have a very clear implications as I talk to you on how you should write your application. Because you should write your application with the council in mind and not the university management, because they will make the final decision on who gets funded. So you have to look at those deadlines, look at the time, what happens, then this, and how long do this thing take? And then the next thing that you ask yourself is, now I have made this, I have figured out all this information. So, and I know what my grant application looks like. And the next thing that you would ask yourself is that, who do you need? What kind of skills do you need? And which equipment do you need to accomplish this work? This is all before you start writing the application. You know this one down. And if you feel that you need a team, and the team here, you're looking at the skills, and I will be talking about where this comes in. So you have to be able to identify, I need the following skills, I need the following equipment, I need a PCR machine, for example, it's molecular biology. And then in my field work, I'll be doing some identification of plants. I'm not a taxonomist, I need a taxonomist on board. I will need a chemist on board. I will need this equipment on board. And then you ask yourself, do I have that already? And if you feel that you have that one and you're ready to go, then the next thing you have to answer is a level of funding. What will the funding cover? This is usually comes out in the call. So, and what can be carried on? What kind of applications are accepted by this donor? Do they accept new applications? The donors will only accept renewals, in the sense that they're saying that if you don't accept any new applications, and others will tell you we don't accept renewals, and so on. So you need to know what kind you're looking at, and then you look at what is the odds of success. What are the chances of success? Because all this determines how much effort you're going to put in. And as I said, putting a successful and a serious grant application needs a minimum of 6 months to a year to do something that is really good. So some talk about appropriations. Like, for example, my experience with the US government is that they'll always say that the money that we are going to grant, that we are going to fund, is determined by 2 things. Number one, it's going to be determined by how much money they get from Congress. So what the budget do they have? Like, say, for example, they can come in and say, okay, we have a budget of $50 million for this call. And in this one, we are going to fund, and then how many are they going to fund? In American system, they always say that we'll fund the meritorious applications, in the sense that they will give out that money until the money runs out. So, and then they don't have a specific number of grants that will be funded. So they simply say, we are putting in $50 million. Each budget is up to $5 million. So if everyone submits an application for $5 million, then we'll fund 10 people. But that's up to. Others will put in a request for $3 million, others $2 million. So that means the number can go up. And so when they say they're going to fund the top 10, then you think about how many applications are going to come in, and you know that that's going to be a very competitive application, and the amount of money that is put in place. So you have that in mind. And then you also look at the budget limits. What are the limits of the budget? And how much, and how is it distributed, and in what period of time? That becomes critical in making a decision. And for how long am I allowed to do this? So this helps a lot in budgeting. And this also helps a lot in terms of how long, and the budget limits are very critical because they determine the level of activities and engagements that you can put in your proposal to be realistic, because that's among the things that will be looked at by the reviewers. So when you look at this one, then once you have this done, then the best thing to do is then you sit down and you draft your proposal. It doesn't matter which format you have followed. You have answered all those questions in affirmative, and you feel that you are good to go. And so the next thing that you have to look at, you go into an evaluation, whether your application is outstanding, excellent, and so on and so forth. So the first thing that you look at is, you come back to the same question we asked. Should it be done? And this is by should it be done, as I said, that look at the importance of research and how do you assess whether an important is research, how do you assess the importance of the research? And the way you look at it is that you look at the significance and innovation. Yeah, is the research significant? What is the significance of the research? And what is the innovation? What new ideas are you putting in? Like, for example, when you look at the research that was coming up, like, if I give a good example right now, if I was to do a grant on vaccine for COVID, would it be significant? Probably not. But if I wrote the next, if I wrote the same grant in 2018, 2019, would it have been a significant work? Now, and then the other thing is they look at innovation. What innovation are you going to do and how are you going to address this? And I'll also go into this one in details on what is looked at in those aspects. The next thing they look at is, can it be done? And what's the rigor and visibility? Again, these are the things that you look at, and these are the things that donors look at when they're reviewing your application. And here, this is focused a lot on the approach. Can it be done? This means just look at the methods. Are you using methods that are accepted, standardized, that can deliver on the desired outcome? If you propose a new method, then you really have to support it very, very well. So because even if what you want to do is very important, but you cannot do it, then you cannot be funded, isn't it? Donors don't fund things that cannot be done. So you have to convince the donor that what you're saying or what you're presenting can be done. And then the other thing is, will it be done? You know, the difference between can be done and there's a reason between what will be done. And I'll give you a very simple difference between these two. Like, say, for example, I did my studies in University of Eldoret, and I've published extensively on use of taxonomic keys in identification of indigenous plants at the University of Eldoret. And then I'm writing this application, and I have all these publications that clearly prove that these methods I'm showing can work, isn't it? But then I'm now doing this work, and I say I'm based in another institution, so I'm in Egerton University. And that's in my new station. And so the reviewer will be asking that, okay, we know from your application that you can do this thing, but will you do it now that you're based in Egerton University? Do you have the appropriate investigation team around you, and do you have the environment? Does Egerton University have that unique equipment for assessment of plant phenotypes that you said you're going to do? So researchers, so therefore, donors will only fund what should be done, and what can be done, and what will be done. So if something will not be done, it will not be funded. And if something cannot be done, it cannot be funded. And if something should not be done, it should not be funded. So the key thing is convincing the donor that what you're doing should be done, can be done, and will be done. And so how do they look at this? So this is so these are the one, and then as you look at it, the other thing that they look at is what will be the overall impact of the project. Will it exert a sustainable, powerful influence in your field of research? And this comes out on the aspect of impact. How will it push boundaries in knowledge? Are you doing what's already known, implementing what's already established? You know, what additional knowledge? Are you pushing knowledge? Like now, like my friend and colleague yesterday talked about the RNA vaccine that came in the time of COVID, isn't it? That one, pushed knowledge. Nobody knew that DNA vaccines could work. But now they pushed knowledge, showed DNA vaccine could work, and now that DNA vaccine can now be used even in developing other vaccine, other technologies beyond COVID vaccine, isn't it? So they look at it and say, how will this impact your field? Will it exert a sustained and a powerful influence over your field? In the sense that you're saying that when I get this funding, should I achieve what I'm going to do, it will fundamentally change a paradigm shift on this. And then the other thing that comes in is that now, when you look at the significance, should it be done? And they say this is, why does the research matter? So these are the questions that you have to answer. Is it important? Is it significant? What's the return on investment on this one? By return on investment here, I'm not talking about from a donor perspective, is what is the impact of your finding? Because if I get funded by, say, the US government, every money that you get from USAID here is US government taxes. Those are taxes, and the donor, USAID, is answerable to Congress. And Congress will be asking, what impact does this change our money do in Africa? So you can come and say, our money changed lives in Africa. So it's never at this stage about that small discovery that you made of that water interacts with chlorine to form something, whatever it is. But more important is, what does the chlorine help human life? So you can talk about, then this one can be used to kill mosquito larvae in water. I'm talking about, supposing we never knew that chlorine does what it does. When you put chlorine in water, it makes water safe for drinking, isn't it? And so on. Supposing we didn't know that, and I'm writing a proposal and they're saying, I've discovered that if you take this element, chlorine, and you add it with water, it forms a form of water that when you put it in, it's very, it's a form that can change the pH of water to acidic and so on. That is not significant to the donor, isn't it? But it starts becoming significant and important when I start talking about what that water does. When I start talking about that water will lead to killing of mosquito larvae and schistosomes that cause bilharzia and so on. And what is more important is when I start showing that what will the killing of the mosquito larvae do? Because when the mosquito, when they die, it's irrelevant, isn't it? Until I start showing that it will reduce malaria, and is malaria a problem? Yeah, then I have to show malaria is killing 78 million people every day. Does it matter that they die? And then I start talking about, when these people die, then this is what's happening. Then they say, oh, then this is really significant, isn't it? Yeah, this hydrogen and chlorine, I think we should fund this research. It's going to improve livelihood in Africa. And the most important thing they want to talk about is not even death and so on. You say, then 78 people will be, student attendance class will increase by 17%, the GDP will increase by 14%. That is what matters. It's not the chlorine, it's the impact of the finding. So many of the time when we write our science, we focus a lot on the chlorine and the water and how they interact and which electron goes around the other one. And then we say this is going to be a very new discovery of a water that tastes like acid and so on. But the donor will ask, so does that matter? So this is where we talk about the returns on investment. And another thing that they also look at in terms of significance is that how will this help America? It's something that many people never think. Donors don't fund you for your own support. All of them. All of them. They always talk about what is the return on investment on the US government. If they fund you to generate new products, how will it help America? And that is why they always say that, and put differently now in a more direct language, they always ask is that, what new resources will this bring to America or to UK? In short, no donor will fund work that can be done in America. So for example, if I come in and I do an experiment on, and this again, a point where we often go off, or we go wrong, is that we propose a research that is so advanced, using techniques that are so advanced, and using materials and resources that can be done in a laboratory, can be available in America. They will always say that this can be done by someone, this should be done by someone in California. Yes, but if you come here and you say, like, I'm trying to look for a new virus, these bats are only found, or birds are only found in Mount Elgon in Kenya, and they could be having another bad virus that can cause pandemic that will kill Americans in the future, they will fund it. Yeah, but if you come and you say, I'm working on this bat that are also available, that are also found in a cave in California, they say, no, it's not adding any value to Americans. What's the returns on the investment? Why should they fund you? Why should they use US taxpayers' money? Always remember, donor fund is not free. It's tied to something. And these people, they're not dishing out money. They're looking at the returns on their own investment. So, as I've said, the first thing they look at that your project addresses an important problem or a barrier to progress in your field. That's what I'm talking about. Is this an important problem? And the importance here is about impact. Always think impact. Do you have a scientific premise for the project? Is this something that you think can be done? And if the aims of the project are achieved, how will the scientific knowledge, technical capability, and clinical practice be improved? Here we are talking about aspect of, we are talking about, and then the other thing that they look at is here, how will successful completion of your aims actually change the concept, method, technologies, and so on that actually drive this field? So they look at the impact. The key word is impact. It's never science. And most of us, we bury ourselves so much into the science of what will happen. But at this particular stage, they're very keen to look at numbers. So it's more of what we term in a usual proposal, what we usually term as a justification. And have you demonstrated the scientific question is the importance of the country or region? So this is usually very important because here we talk about the issues of relevance. Like if you're talking about, let me give an example, Ebola. Someone is doing research on Ebola in Egypt. I'm picking on Egypt as an example for the simple reason that Egypt has never registered any case of Ebola. And somebody else writes the same grant of Ebola in Congo. Who do you think will be funded? Is a person in Congo. So wherever you're writing, you also have to emphasize on what is it, is what you're saying, is it a problem in Kenya? And this is now where you have to go back and cite government documents and say this cabinet meeting of this day prioritize this one as a major problem of major importance is in Vision 2030 of the government of Kenya, so that it fits in, so that it appears to fit in within the paradigm of the local research that's going on as a part of the government, because you don't do research and implement it in isolation. And this is now where you introduce government institutions. If you're doing research on health, you involve a local dispensary, a hospital. You say, these people are dying in this local hospital, this will address this problem. So you have to domesticate your application and make sure that whatever you're doing is relevant to your country. The other aspect is innovation. This is again looked at independently of significance. There's a very big difference between significance and innovation. Innovation is about novelty. What is new? What I talked about earlier was not about anything new. What I talked about earlier was about the importance of why you want to do what you want to do, isn't it? But now this one looks at how do your application actually challenge or seek to shift the current research or a clinical practice or a paradigm. So you have to show that whatever you're doing has not been done. And this, or that you're employing a new method or a new tool in your study that has never been done before. Like, say for example, I can come in and I can talk about, now let's talking about plant taxonomy. with a good reason. So supposing I come in and I say, I want to do plant taxonomy. I want to look at the plants in this region. And in this particular plant, I'm getting into a forest. And then I say, I'm going to use the taxonomic keys that are well established right from the beginning, which are what we call the classical tools that are used for, those are known tools, isn't it? So if I say I'm going to use these particular tools for classification, identification of these new plants and so on and so forth, they'll always say that, well, that one will work, definitely, isn't it? But it's not novel. Because they say these tools have been used for many years to do the identification. They work, we have no problem, but you're not shifting, you're not pushing the boundaries of knowledge in that aspect. So, but if I come in and I say, like, in addition to this one, I'll also use the barcode of life. I'll get the DNA, do some things on the DNA, use next generation sequencing, a new tool that has not been determined, and I will also employ this new AI, artificial intelligence tool that also looks at the stems. All the methods are usually based on the flowers and the stem, but this new method will also look at the root, and then it will be able to do this. This method, if working, will then be used to characterize all the trees in Mau. They look at it and say, this is novel, isn't it? And they say, should this new method that this person is employing work, it will have a very broad application in a taxonomy across the world. Then that looks novel in a way. So they look at it and say whether it's novel to one field or in a broad sense. So this can also be in methods. And then they look at it if it's a refinement or improvement of a theoretical concept and so on, intervention. So this is where many people get confused between innovation and incremental knowledge. Incremental knowledge, most of the time, don't get funded. And so they look at it and say for example, going back to the aspect of taxonomy that we talked about, I come in and they say, I'm using these methods that he developed earlier, which are now documented as novel, but they're now a standard mainstream, they worked before. But what I'm doing is that now, he did this work in Eldoret, isn't it? And say in around the forest here in Eldoret, but I'm now going to use the same methods in Mau Forest. And so in Mau Forest, there are other different species that are there that are not in Eldoret, so I'm going to use these tools. Then that is considered as an incremental knowledge. It's not novel. You're just adding to knowledge of what's already known. You're just expanding the information. So that's something else that you look at. So we come to what is, should it, now you're coming, can it be done? Can it be done? So here they look at your approach. The method is a method that you are employing. What are your plans, methodologies, objectives, and aims? Is the method you're using, can it deliver on what you want to do? Like say, for example, I'm trying to look at I'm trying to measure distance. I'm giving an example. I'm going to say something that's very different completely. I'm trying to measure the distance between here and the gate. It's part of what we are proposed to do. But I'm using liters to measure the distance. Then they say, I'm using beakers of water to measure the distance. And then they say that, that can't work, isn't it? As much as the idea was very good, it's something that this measurement of the distance between here and the gate is very, very important. But the tool you're using will never be able to give you the distance because you're not using the right tools. And the other thing is, can it be done, comes in the issues of, how are you going to do this evaluation assessment to ensure that this can be done? And is the financial resources are sufficient to get this done, the money? So here they look at your overall strategy, methodology, whether it's well-reasoned and appropriate to accomplish specific aims. So they go back to what you want to do. And they look at the tools. Are these tools capable of achieving this goal. That is where we say, can it be done? So they look at aspect of analysis. Do you have a robust and unbiased approach? What we mean by this is that, like, let me take, for example, I'm going back to my plant story. So I've done this taxonomy, and I've used this particular taxonomic key, and I've identified that that plant is one of those difficult botanical names. So I have it there, and they say, okay, using my taxonomic keys, I'm giving an example. Using my give me one botanical name, just Biden spilosa. Okay. So I have come in, I've used my taxonomic keys, and I've identified that this plant that was unknown is Biden spilosa, isn't it? And then the next question is, how sure am I that those keys are working? Here we talk about the issues of robustness. So I have to indicate another line and saying, okay, I have to come up with a validation system where I'm saying that once I've identified that this one is Biden spilosa using the keys that are published everywhere, I'm going to take these samples to Kew in Germany, to another taxonomist in the University of Nairobi or elsewhere, or the Museum of Kenya, who will reconfirm based on their keys. And additionally, I will also go down, and because the genome or the genes of this plant are known and they're published, I will also get the DNA, look for that particular gene that is used in taxonomy, and then confirm again. So here we are talking about robustness, isn't it? That I have used 4 different methods to confirm that I'm on the right path. And my approach was unbiased. You know, when I went to collect this tree, I made sure that what I have was representative of the tree in that particular area. And this one, therefore, I'm very sure that I've put in what we call a fail-safe system, where I'm saying that should this one fail, this one will work. Should this one fail, this one will work. And so I've also looked at the potential problems that can come out. For example, I will indicate that, okay, I have realized that I've indicated, you write, and you say, I've indicated that I will get the validation of these markers, or this using the National Museum of Kenya, or herbarium, so the herbarium, the non-herbarium in Kenya, which have type specimen of this plant, for which they can be able to compare and confirm that actually what I have is Biden spilosa. But should they not be available, or should there be a riot in town and I cannot be able to take it to the lab, I will go to another lab in South Africa that has accepted. And then I have a letter from that lab in South Africa stating that should there be a problem, my lab will be available. So what that means that I should come up with alternative strategies. That I need to look at the proposal again with the probability of failures, and I identify all the areas where this proposal can fail. I say it can fail here. This person may not be available. You know, University of Eldoret, but decided to say they are not taking in my sample for analysis. Like, say, for example, I say I'm going to analyze the samples now in the very good laboratory here for molecular biology. But what if these people say, no, we are not going to analyze your data? You remember the story of the bank? The bank say you have to come up with a tight business plan. If the bank have the slightest fear that, you know, that your approval, for example, you want to build a house, if the bank has a very slightest fear that your approval for constructing your house will not be approved by the council, will they fund you? They won't. So the same thing, these people also come in and say, should that happen, what will you do? Because the challenge with the donors is that once the money has been given out, it's very hard to retrieve it, isn't it? So they're trying to see here, will it be done, and what are your benchmarks for success? How can you be able to tell me that you're on path, you're on route to achieve your desired objectives? So you have to give me some very clear benchmarks that I will assess I will compare my performance. I will know I'm on the right track by comparing my finding with X, Y, Z. I will use this one as a benchmark that if I do this, I'm on the right track. If I do, for example, with the benchmark would be number one, I would come in and say, I'll look at those plans and so on. And the other thing that also comes is that, well, will you establish the feasibility, and particularly the risky aspect be managed? What if something goes wrong? How will it be done? And feasibility is very important. And more importantly, do you have an adequate plan to address relevant variables? So this is about robustness, convincing the donor that what you're doing here, your plans that will address every such variable that comes out, and what is your collaboration plans? Who will do what? In your management, collaboration plans here, we are talking about your management plan. Who will do what in your project at that particular time? What will be the role of who? In short, give us your organogram so that we get to know when things are going wrong, who needs to be, you can be able to identify who to pull out of the project. When things are not moving as a PI, you come out of that. And the idea of feasibility, the most important aspect of feasibility on this, can it be done, usually comes in what is termed as preliminary data. That's why it becomes very, very critical for every donor. They want to know that, the question they ask is that, are you trying to do these things for the first time, or have you ever done it before? If you've done it before, then you can do it, isn't it? If you've never done it before, then we don't know whether you can or you cannot do it. And the only way you can show me that this can be done is show me some preliminary data. But in preliminary data where you say, we are encouraged to do this thing because our preliminary data shows the following. In the preliminary data we use this method that we're describing, and we got some results. But these results are pointing in this direction. That's encouraging. And this result is what is now informing us or encouraging us to take this direction. Because the point is, show me you can do it. That's critical. And the other thing is, still on this, is that what is the likelihood that your collaboration in advanced development of local scientific capacity, build local research infrastructure, or increase collaborative research and a partnership? Yeah. You have to convince me that your collaboration will also advance local expertise. And that is why in almost all good grants, you have to put in what the term as a capacity building component, where you say, we are going to recruit a master's student or a PhD student to do this work, that capacity building. And then talk about how will it advance the collaboration. You say, this will develop foster collaboration between University of Eldoret and Kenya Marine Services, where we will now be able to work together to develop new methods, and that will be addressing areas of common interest. So we now come to the last bit here, where we talk about, will this investigation be done? Do your investigators have the skill set and experience to deliver? So here we are still looking at, will it be done? Now you see, we said, should it be done? We have seen that, can it be done? But the question is that, will it be done? Isn't it? Because as you say, if it will not be done, then it will not be funded. So will it be done? So here we are looking at aspect of investigators. Are these investigators, do they have the skill sets sufficient for you to deliver on what you want to do? And this is now where you come in and you select who are the best. You look at the skill sets, like say, for example, if I look at the aspect of the plant, and they said, I want to look at the taxonomy of the plant in this particular region. So at least among the people that I will need, I need a plant taxonomist. I need someone with extensive experience in DNA work, isn't it? I'm just still looking at people. I need people who have an experience in DNA, plant, and so on, in extraction and DNA. But that's not just, that's just here. Don't forget that we want to do this work in Mau, isn't it? So for me to get access to Mau, I need to have someone in forestry. this is a question of will it be done? Because like say, for example, I read a very good proposal. I've shown it should be done. I've shown it can be done. And then I said, I'm going to do this work I'm going to collect these plants in Mau Forest because of the subject of my area for this very important herb that can be used to cure all the diseases. I need to identify it, and then it will solve the problems for the world. And then on the day that I'm going to go to the field, the people in Mau tell me they cannot give me an access permit. That's it. The entire thing dies there, isn't it? So if I go to Mau and they tell me, you cannot access our forest. So I have to make sure that in my application, I have a letter, or I have somebody who works in that forest. And this person from the forest will also give me, we are still looking at the skills here, he will tell me that he's very familiar with the terrain of that forest. Because they tell me that, okay, what if I go to the forest and I have all these ideas and I've never been to that forest? Where do I start? These are now the questions donors ask. Supposing you get there, you are the entry of the forest. And then they open the door for the forest for you and they tell you, okay, get in. So where do you start? So I must indicate here among my investigators that I will be working with the chief forest officer in Mau, who has some background training in plants, and who is very familiar with the terrain in Mau, who knows pretty much an idea about where each plant is located in the Mau forest, and who has a map, and who has done these 3 or 4 publications on flora and fauna of the Mau forest. Isn't it? So that person will hold my hand when I get there and take me around and show me this is where the trees are and so on. So they look at whether your team of collaborators are suited for the project and whether they have appropriate experience and training and all this. And if they are training, if they are early stage, you remember you said early stage investigators. And if you are accomplished, they have demonstrated ongoing record of companies that have advanced their field. So once you're considered as an experienced investigator, they want to see your records. If you're an early stage investigator, as I said, they only want to see, are you really trained to characterize plants? But if you're an experienced researcher, they show you, give us your publication record. They want to see how far have they gone. And by publication record, they'll be looking at the entire group. And all this, how do they know this? This is where they always tell you that attach for us a CV of the participants. Something that people do in a rush, isn't it? Without customizing those CVs to suit the application. So when the reviewers look at the CV, it has nothing to do with this application. Yeah, they look at it and say, well, these investigators, none of them seem to be suitable. And it's based, and then you start complaining, oh, that guy is very good. I don't know why they're saying this, but the person sent an outdated CV. Yeah, and then that CV has to be customized for that particular, and they'll always tell you how they want you to write it. So this is used to look at the investigators. It is never about anything else. So the others, they ask for CV in many ways. Others say, give us a biodata. Others say, of the investigators. Others will tell you, give us a bio-sketch. Others will tell you, give us a description of the investigators who are involved. And this is a bit that people write in a rush because it's like the last thing. And remember, I talked about the issue of a deadline. These are the things that Donald will send to you, for example, the last minute, when you have been bugging him for the last 6 months. And then the last minute, he tells you, oh, yeah, take it. And then you take it and you upload it. And then say, whew, done. Yeah, when they get in, they say, okay, where there's a place where I say, should it be done? There's a place where I say, can it be done? Can it be done goes to the method, isn't it? And that particular method is a point where I said, Donald will be doing the identification of grass of the roots of the plants, isn't it? Now, they take that bit and they take his CV. And they want to see only those words. where he says, I have a lot of experience in plant identification, including the roots of this tree. And I've done this root of the tree. They're talking about not everything else he has done, but they're only talking about that root of the tree. He needs to write a paragraph to justify why he feels he can do that work. But because he sent, or I sent to him my generic CV, which he uploaded, the CV is very big, but does not have much weight on that aspect. So they look at it and say, I don't think this guy can do this work, isn't it? That will be the assessment. And so when the reviewer comes back and when the review report comes back and says that I don't think he can identify this plant, he hits the roof, isn't it? And say, how come they tell me that I cannot? I've been doing this work. But you see, all this is based on what you submitted. You're given a chance to present yourself. You didn't. You've presented information. So that bit for the PI, someone who is reviewing, what we typically do is that we review those applications very well, the CVs, and we rewrite them. Because I'm the one, like, if he's the one who is a PI, he knows what he had in mind when he recruited me in the program, isn't it? So I write to him and I give him my CV on there, then he can sit back in it and rewrite it to suit what's in the primary part of the body of their proposal. And you see, mistake is many people tend to focus a lot more on the science, but they forget this one. And this one is called independently from what you have seen. Because if it will not be done, it will not be funded. So, the other thing they look at is whether this has a complementary and integrated expertise. This is usually quite important, and this usually comes out through what is called co-authorship. What this one tells us here, by just looking at your CV, all these things I'm talking about will need to be done, the investigators, all this information is drawn from your CV, what you submitted. They look at it against what's supposed to be done. And then the other thing that the donors want to know is that, is this a consortium that has been cobbled up in a rush just to get this money? And once the money comes in, then people start fighting one another because they don't have chemistry. They have never worked together. And so how do I know that? We go back and look at the publications. Have these people co-authored the papers? Have they worked together? For the part, and that's we talk about the complementary and integrated expertise, where we say that these people, each one of them have their own strength, but they overlap, and they've been able to work together. And is the research team as proposed likely to develop scientific capacity and infrastructure in participating institutions? So once you have that done, then we come now to the environment. Now we have moved away from the people. Will it be done? So if I say that this work will be done, the environment, where will this work be done? Environment comes from institution. Does the institution have appropriate tools, appropriate skills, and appropriate, not skills, but appropriate equipment? For example, I indicated somewhere that I will use a PCR machine, or I will use some to do the DNA extraction, isn't it? So this is now where most donors will always ask you somewhere that, just give us a one page description of the resources in your institution. Resources. And then this is another one that is usually done in a rush, where, you come and you say, I just want to list 1 or 2 or 3, because what is important is the proposal. Because you are made to believe in your mind that it's detached from the proposal. Yeah, they tell you, give us a list of resources in the University of Eldoret. And then you say, because I'm in a rush, I will only write what's in my lab. Yeah, what's in that lab? I don't care, I don't like that person. I cannot put the equipment in his lab. Yeah, so they come in and they look at it and say, we don't see a PCR machine here. Yeah, he indicated that he will do a PCR machine. He used a PCR machine, but the list of resources that have been given here do not show us anything. And then they look at aspects to do with the population. You say that you're going to do this work in Mau Forest. That's another environment where this thing will be done. We don't see a clear description of how they'll get to how they'll get to Mau Forest. How? Because we can see the budget, he has not put in a budget for a vehicle, but in the list here, they have not indicated that the university has Land Cruisers and Land Rovers that can get into Mau Forest. So we are not convinced that these people will be able to go to Mau Forest. Because it's not clear to us how this team will move from University of Eldoret to Mau. And then, so here we look at the scientific environment, which work will be done, and how will it contribute to the probability of success? Is it likely to be successful? That the environment is sufficient to provide this? And is institutional support, equipment, and physical available to the investigators? And this availability, the keyword is available. The equipment can be there, and so on, but it may not be available to you. That's possible, isn't it? Yeah, and that's now where we want a letter from the VC. That is what we call the letter of support. Where the VC indicates that this equipment will be available to the investigators. So that should this grant fail if this one does not work and the investigators are unable to get access to the equipment, then we can sue the VC. You know, the investigators can take the VC to court, isn't it? Because we will say that the VC committed the institution by stating that this equipment will be available, so they have to make it available. Or they go to court. And then they look at what are the unique features? With the project benefit from unique features of scientific environment, subject population, or corroborative arrangement. What you mean by unique features here is that, for example, we are talking again about the issue of the Mau Forest, isn't it? Then you find that this Mau Forest is a unique feature in terms of ecology of plants, isn't it? So how will this benefit from? Then you can say that this Mau Forest has very unique features that are only present in the world. There's this particular geology or plant and so on that only grows in this particular area that anybody else in America cannot be able to do it. They have to come to Mau specifically, or the population, or University of Eldoret has some repository that have been collected in this region from 1942 that is only unique to this place. So you have to come and say, this is now the environment. The point is, why should it be done here and not anywhere else? So you're saying, here we are robust new technology, or you can say, we have equipment. We are the only ones with equipment that can measure radius of a distance from here to wherever in an efficient way. So what are unique features that are there? And once you've done this, now you've looked at your proposal, you have addressed all those issues that I mentioned, isn't it? You have shown that it should be done, you have shown it can be done, and you have shown it will be done. Now you start thinking about who are your audience? Who are the audience? You only have two audience. You have the reviewer, and then you have the board. You remember we said the reviewer will only review and take to the administration. The administration will look at the review report, and based on the review report, recommend for funding to the board, funding board, isn't it? Like say, for example, if it was the University of Eldoret, this is then forwarded to the council. It's the council that finally decides, not the university management. The management only puts together the responses and scores them, isn't it? And says, okay, out of this, because the management will say, oh, we are going to fund 10, so give us the top 20. So that's now the board. So the management looks at these 500 applications and picks up the top 10, the top 20, takes it to the board and tells the board to select the best 5 and the best 10, and these ones. Now, the key important thing here is that who are the members of the university board? It's just the vice chancellor It has a representative from Treasury. You can see how diverse it is becoming. It has a representative from Ministry of Education. It has maybe a bunch of politicians on board, and so on and so forth. But those are the people who will decide. And so, if they cannot understand what you wrote, it will not be funded. So, in that particular case, you really have to look at the significance and address the shortfalls and edit the proposal for your audience. This is what I mean. So you have to be able to convince the experts in your field and a mixed audience from scientific background and prepare your summary for a lay person. This can be a deal breaker. And that's what I'm talking about. Because the person who comes in from, who flies in from Nairobi, from Treasury, to attend that meeting, don't forget that his vote, in that meeting, the Vice Chancellor is only a secretary. And in that meeting, the Vice Chancellor, he's only person who has a technical knowledge of what is happening. But unfortunately, he's the secretary. The other decisions are made by the chairman of the board and the members of the board. So look at this person who has come in from Treasury. What will be his background? It's maths, isn't it? And GDP. And so he's sitting there and he's told that, and he's told that, we have these 20 public applications, but we can only fund 10. So you from the Treasury, you want us to advise us on which 10 we are going to fund. And this person from Treasury, he just took the flight in the morning. You see, all this document was sent a week in advance. But during the flight, he came in the office and he found the driver waiting for him outside, telling that, the university are waiting for you. So he takes this flight, and during the flight is when he starts reading through what he can read, and he reads the summary to get an idea so that he can make a meaningful contribution to the discussion. So obviously, the ones that are too complicated, he just keeps them away, isn't it? And the ones that are easy to read are the ones that are changia sana kwa mkutano. When he goes, oh yeah, this one is good, so that is and so on. So that one comes in. The other thing that also comes in here, as I said, you avoid jargons. So you make it for the lay person. Number 3, the other thing that you also have to do is that now, at that particular point, all these 20 applications, don't forget that they have all been approved by the board for funding, by the management, that all of them can be funded, isn't it? So you're not talking about scientific merit anymore. All of them can be funded. So this is now where other considerations come in. And the most important thing that they look at is the gender. They want to see, is gender mainstreaming being captured in these applications? Who are the collaborators? And it is for this reason that sometimes when you're putting together your team of collaborators, you try as much as possible to include as many women as possible. That can be a tiebreaker. I was reviewing a grant for the South African National Research Foundation, and it's called the score sheet that they gave me. Part of it was how many women are involved. And there's a marks. 10 out of 50. So the very good proposals made up of only gentlemen, 0. And don't forget, at the end of the day, it's the score that matters at that particular stage. People look at it. So you have to think about gender mainstreaming. And these people also look at aspects of this regional balancing. Remember now the science is out. They're looking at issues of regional balancing. They're looking at it and say, is this a really big problem in the region? Or say, last time we funded this work on malaria. Now, I think we have never funded this area. So you can see how the whole thing goes. And then from there, they come up with a list of priority. 1, 2, 3, 4, 5, up to 10. Then they go to the budget. They now go to the budget committee and tell them, distribute this money across these people on their budget, because you have a budget of, say, 20 million shillings or 100 million shillings. Distribute this money until the money runs out. Or give the first 10 if the budget was fixed. And then they take the top 10, and these are the people who are funded. So who finally decided who's going to be funded? is the board. It's not the reviewers, and it's not University of Eldoret management. And that happens across all donors. And I mean all, not some. The work of that people who write to you those things, respond to the call and so on, the job is limited to just collating that together and ranking them. And when they finish ranking, they push this to politicians to decide who is going to do. So you have to make sure that you avoid jargon, write a thing in a very thing, and then of course you can always have it proofread by a colleague. And usually when you send it to be read by a colleague, don't argue with the colleagues. When they say they don't understand it, just know that it's something that is not clear, so you need to work on it. And finalize the budget. You finalize the budget issues with the institution, because the worst part comes in when you get funded, and then you find it does not align with the institutional policies, isn't it? The institutional policies of overheads, what can, allowables and allowables, because it's the institution that will actually be implementing this grant. And then you submit the clean version of your application. So here they come, and they give different scores. So what I'm trying to show here is that each part is scored independently. While most of the grants that people write, they focus a lot only on significance. That's what most people do, the science. And maybe some aspect of approach. But they never bother much about investigators. And in most cases, they usually indicate that I will work with John and Peter, but I don't indicate their role in the application. And others don't care about much of the environment. When that small paper you are told to give us a list of resources, and then you write it in a small corner in your office, alone, in a rush, and then you upload it just to fulfill that requirement. But then you come and you see that the scores are actually based on, each one has an equal weight. And that's why you find that you wrote a very good proposal and it was never funded. Because the investigators, you could not convince them that the investigators could be able to do this. The innovation aspect, you never gave much interest. Approach, you wrote it in a rush. You never looked at it, you never made it a fail-safe. It was not robust enough. The rigor was poor. And the environment, you say that you'll do PCR, but it does not show anywhere that your institution can be able to do this work. So you have a problem. And more often than not, from what you can see on a score of 1 to 10, 1 being the best and 10 being the worst application, most grants will only fund those who get 1 and 2. 1 being exceptional and 2 being outstanding. Anything excellent, very good, anything like excellent, very good, and good are not good enough for funding. Yeah, your grant has to be exceptional or outstanding, period. Excellent is not good enough, and very good and good are not good enough. So, reason why you really need to, and that's why it takes a year, 6 months to a year, to put together a very good, successful grant application. So, what if you're not funded? So sometimes some months give you room to resubmit, and one of the things that you do is that so you write a very clear, something comprehensive rebuttal. In most cases, you address every issue that came up, and you acknowledge and address also of weakness that described in the feedback. Now you're doing a resubmission, and you clearly explain the disagreement and concepts of accommodating some of the feedback, because you are now trying to implement some of the comments that you got. And you try to be, you don't come out as very overly defensive. Yeah, you try to be a little bit civil in your language, where you begin by saying that, while I agree with this statement, this concern, so you start by, the statement is an agreement, isn't it, with what was said. But then you come and you say that, and then you say, I just have a little concern about this statement. Well, what if you get funded? So funders' goals are usually identify that the relevant is research, the research is relevant, fits the research mission, as you have said, and is impactful. Remember impactful, where we talked about the issue of that one. And then another thing that they also look at is that there are very many ways you can also get from others, and that you also have to understand that all different reviewers, all different granting organizations, they use the same criteria in review. It doesn't matter, by the way, whether you are sending it to, I'm talking about serious grant organizations, whether you're sending it to Wellcome Trust, you're sending it to NIH, you're sending it, even if you're sending it to Ukraine, same things will come up. It's only that the only difference comes in formatting, and it also comes in how they ask those questions. They'll always ask them in a different way, but they ask the same questions. Others will come up front and tell you, break it down into innovation and so on. Others will tell you, just tell us what is the importance of this and how does it help. And so you'll get funded if you read and follow the instructions, the call, if you pick an important research question, if you use the right method. You remember we talked about if it can be done, if you write in a very clear account, in a tight structure, in the way you communicate, and if you did the application with the reviewer and the audience in mind. So, coming up like an overview of this, we find that if you generate an idea, so it usually begins by you generate an idea where you know it's novel, what can be done, then you find a matching funding opportunity, and once you come a funding opportunity, which you look at in different ways, you do a background research to identify that you have all the tools that are necessary for you to do this one. You read the technical aspect, the technical portion, you check the administrative parts, budget, institution, what do you need, are there letters that you need, and one of the challenges that I've had throughout in my grant applications has always been one, external letters of support. So you have to give people enough time to get them to you. This is usually the most challenging bit in all grant applications. It's not the writing of the grant. The hardest part, again, I have to repeat this almost for the 10th time, is getting letters of support. Because this letter of support must come from the highest level in that institution. Like say, for example, if I am collaborating with the University of Eldoret, the letter of support must come from the Vice Chancellor or the Deputy Vice Chancellor. That's the lowest it can go for the donors. Because the letter of support must come from someone who can commit the institution. And so if you're doing, and I'll show you in an example here where you have so many people, you have a letter from each one of them, and you also have a letter from individuals, where you also have to have a letter of commitment from individuals. Because I cannot put her in an application without her knowledge. So she also has to write to me a letter committing that she'll be able to accept. And then she also has to give me her CV. And she also has to give me her bio sketch, indicating that, and that's just one person. And then if I have 15 people, and I have around 10 institutions, and then I'm waiting for the last day, and then when I'm asking people, can you get me a letter of support? Then they tell me, oh, the Vice Chancellor is out, he's in Germany. We have to wait. And then I say, oh, I need this letter. And yet the role that I said that University of Eldoret is going to play is so critical, and it's just a day or hours to submission of the application, and I cannot remove the University of Eldoret. And if I submit it without a letter of support from the University of Eldoret, this grant will not be funded. So that's usually the first thing that we do typically when you're writing a grant. We identify partners. That's why I said that before you write. Remember, I said before you start writing, you identify the partners and you start telling them early enough, and I'll tell you how this one works. In some institutions, and one of the reasons why in Africa we are a little bit challenged, is that in other institutions in the West, they have an office that is called Office of Sponsored Projects. Others call it Grants and Contracts Office. And the work of the Grant and Contract Office is that it relieves the scientists from that work. For example, if she's doing an application and she's seen this call and she responds to that call, this office takes over on admin issues. They only ask her, who are the people you're going to collaborate with? And she gives a list, and email addresses and phone numbers and contacts. And then she asks, what do they need? What do the donor need? He will not tell them. She will go to the website and look at the donor wants 1, 2, 3, 4. And then that Office of Grants and Contracts takes over so that she can focus on the science. This is that office that will be writing to these scientists, give us your report, we need this, give them deadlines, send them reminders. We need this thing done by this time. The same office will also look at the donor, budgets, requirements, contract with the accounts office in the university, come back, prepare the budget for the PI to review and say who is coming in, what are allowables, non-allowables, is this work, who is coming in, who is not, distribution of funds and so on, so that the scientist focuses on writing the proposal. And then finally, it is that office that submits the application. So once he is through or she is through with the application, she only sends it to that office and says, I'm done. So you people do your job. So these are the people who now takes over this application, logs onto the donor website, uploads all the information, make sure that they're all in compliance and is done, submitted on time, so that she can start writing the next proposal. So she can keep on writing proposals. But in Africa, we do everything. Where you write this, you go, you camp in the office of the Vice Chancellor, I beg you, give me that letter. Oh, that letter, and then you're telling you I'm not available. People are not picking up your calls, and on the side you're trying to write and you cannot write. The people from finance, they come in and say, kwa nini hujaniweka? Mimi ukaniweka, mimi hiyo pesa sitafanya. The policies. But you see, if there's an office that does that, it's a policy. They simply say, this is how it's done. This is a policy. It's not negotiable. You agree with me, isn't it? But in Africa, I saw this, and then when the grants are awarded, they're also called post-award. It's also handled by that office. The same office that the grant, the people who dealing with reporting to the donor, is different from the university finance office that runs the day-to-day of the university. Yeah, but this particular office now deals with the donor issues. If the donor comes and says, we need a technical report by 5th. We need a financial report by this time. Oh, there's a new change in the donor policy that everyone must send data to this person. So this is the person who informed the researchers that this is in this new. But this office, when this runs for a very long time, it gets to know every donor. That office, Grants and Contract Office, it knows every donor and the requirement well in advance. So if they see another call, oh, that one is from USAID, we know the rules over time. And then they'll also be fishing out for calls. That's the Grants and Contracts Office. In America, all universities have that office, where they send, so that the scientists focus on writing the grant. The Grant and Contract Office does the other paperwork. Then scientists can be able to do many, many applications at the same time. So here, again, is where you can now come and you submit, and you forget. And so, if you look at the time distribution, you find that actually looking for a call usually takes around 20%, 25% of the time. Planning the proposal, 25%. Writing, only 35%. Admin and submission takes 5%. So many people think that writing takes almost 70% of the work. No. The most important thing, and the best grant that can ever be written, is one that is not written. So that's why we talk about issues of success rate, isn't it? Success rate is how many grants you submitted and how many were funded. So the point is, you should look at those points that I've indicated up there before you start writing. If you feel that the work you're doing should not be done, don't write it. If you feel that this thing, cannot be done, don't do it. And if you think that it will not be done, don't do it. And that is why sometimes we get calls. And I can, as recently as yesterday, I got a call from my colleagues in the US. There's a grant that had just come out by USAID for around $35 million. I got an email, I think 2 days ago. And from my American colleagues, and they're asking me, can we do this grant? And within 2 days, we realized that we cannot do it. Based on that criteria, yeah, we said, no, we can't. And we don't do it. And there's the one that comes in and we look at it and we say we can, we should, and we will do it. Then we embark on it. And within some time, maybe within 6 months to a year, we submit and we get funded. Simply because we do a lot of that search. So what you're trying to avoid here is a situation where we submit applications for the sake of submitting. Where you come in like a university and say, oh, this is an NRF call. Everyone submit application. Then people rush to submit. Everyone submit. Then you have, oh, we have submitted 17 applications, 2 are funded. But had we just sat down and said, okay, let's step back a little bit and look at each application that's going out. You can look at it and say, wrongo. This one will not be funded. Why don't you spend your energy doing something else? You get me? And then you send a few that are very competitive. And then those ones you work very, very hard on them, even at an institutional level, as we said, so that it gets a point when donors look at the success rate of grants coming from University of Eldoret is 90%. Like me, in my group, you have a success rate of 90% of the grants we submit. 90% are funded. But it's not that we wrote, we don't, there are very many we don't write. Because we look at them and say, here we don't stand a chance. Because we don't have this, we don't have this, we don't have this. We don't have the right party. This does not fit in very well with our theme of the skills we have, the skill sets. So I don't think we will be funded. So that saves us time. And so remember that you'll always assume, whatever you write, always assume any problem that your fault is not that of the reviewers. So if it can come up. And if reviewers have misunderstood something, then you did not explain it clearly. So it's never about them. And make sure you invest considerable work and effort in the revision. Reviewers will do the same. And then finally, you submit and you have some good luck. So quick tips: understand the funders, recognize the share evaluation, know what's really all the reviewers that they look at, that's really all the same review. So whichever way you go about it, use the same criteria as your roadmap, the one I've given. Use the key questions as a guide. And keep time, check your style, know your audience, connect, recycle, and then be original. So we have used this tactic to get around $9 million so far. So I'm just talking about the outcome of the same tactic that we have used. So we've had this grant from all these different donors, close to around $9 million. And so we are now spreading the gospel on how it can be done. So we got some funding from the US government to do this work, where we are now training, empowering early career researchers in East Africa. And we're also developing leadership and institutional capacity to disseminate knowledge in this innovative system. The idea here that we discovered is that in Africa, most young PhDs are never trained on how to get money. The universities are probably some of the most notorious because the universities hire the best, the cream of this country. But once they're hired, they're not usually trained on how to hunt, on how to get money. The next thing is that get money. So we approached the donor and we said, you want to bridge that gap. So we are training postdocs. We identify PhD students in an institution like this one, young junior faculty, and then we buy their time. We come to the university and we say, we want to buy their time. We pay their salaries for 2 years. And then we train them on how to get money. They get money, they establish their labs. And then we're also getting middle-level managers and taking them to the US to Yale University for around 6 months training, on didactic training on how to manage grant. Because we realize that another part that is really lacking is that bit I talked about of the grant management office and how to help people get grants. So that's the bit that we are doing. And we are doing this as a collaboration, and this now, also the grant that we wrote, It involved all these institutions, from you can see KALRO, Nimri in Tanzania, KEMRI, Yale University, Maseno, TVLA in Tanzania, Gulu University, CDAIM in Kuala, Colombia, and MUST. So you look at it, and this I'm talking about letters. So to do this, we had to get letters of support from all these institutions. And then we also had to get letters from these individuals. Here I'm part of I'm the PI and the lady up there from Yale University and ourselves. This was our executive committee, so they had to come in. And then you also had to get a letter from advisory committee. These are different individuals from different institutions, like from what you can see from ISTECO. Some faces, I believe most of you are familiar with them, like Rosemary Sang and so on and so forth. And then you also had to get a multitude of letters from from mentors. People are going to serve as mentors in this program. And each one of them had to give the same letter. So you can see it takes effort to ensure that all this come in good time to do this work. And then you also have to come up with how we are going to do it, where we said, we have to have the virtual courses, we have training, recruitment of postdocs who are going to come in, and we have successfully recruited for our first group, the 4 postdocs. We have Edith Chepkorir in KEMRI, we have Tekla Angelo in Nimri, in Mwanza in Tanzania, Geoffrey Walakolach in Gulu University, and Benson Mashira in Pwani University. These are people from whom we have bought their time from the employers for them to build up their careers. And then you also have a lot of virtual training on disease, on virtual training on disease seminars, where all the people showcase their work. And finally, we have series of workshops that we are now putting up for skill building for 5 years, ranging from grantmanship to biology of disease vectors and so on. Skill building things that we are bringing to address this shortcoming in the region. And that's it for now. And what if you get your money and you publish, then I can come and tell you how to write your paper and get it published. That's where we are.
Thank you, Paul, for that very insightful presentation. And I believe as researchers here at the University of Eldoret, we can see the areas where we come short. I think to me, it's very clear. And very quickly, one area that I see is we get calls maybe a month to the deadline. And now he has told us they usually prepare for 6 months. So I think that can give us an understanding of why sometimes we don't succeed. If I get a call today and the deadline is three weeks down the line, and I have to get all these letters of support, I have to go through all those steps in preparing the proposal, and I still have my teaching responsibilities, administrative responsibilities, you can just see that I've started failing even before I start. So I think these are lessons we should learn and see how we can readjust ourselves so that we conform to this method or this approach of writing grants for funding. So, Paul, we thank you so much for this presentation. And I believe we have some questions, some comments we'd like to make. So I want to open it up. We'll start with 3. Yes, we'll start with Prof. Luther. Do you have a mic that I can use? Anybody else? Yes, we have another Professor Gohole. Anybody else? The third one? Let's start with Professor Luther here. It's not on. The mic is not on.
Okay, thank you. Okay, thank you, Paul, for the presentation. I think it has been a learning process. I have some, maybe, I don't know if there are comments or questions. One is, I know you are in the academic sometimes back, you are in Egerton, and now you have moved to a research institution. I know one of the challenges that we face, maybe as a country, is we have a disconnect between the academics and the research institution. And maybe from my experience, I don't know how that can be addressed, because it seems also to be affecting us in terms of research. And then number two, you have talked about the training opportunities, which I'm interested with. As the Director Board of Postgraduate Studies at this university, I would just wish maybe you try to highlight on that, and also tell us how we can be able to get that information so that our students can also benefit for the same. Thank you.
Okay, Professor Gohole.
Thank you very much. One thing I would like to ask is the way you are just saying that sometimes you get a call 2 or 1 month to the deadline. So in this case, would it be advisable to start having ideas, putting them together, although that takes quite some time, and then you just readjust them when you get a call that matches what you're writing? It's like that now you start preparing early enough, and then by the time, there are some places, there are some funders who give out calls at certain times, and generally you would have an idea of what they want to, what the areas that they fund. So start preparing early, get your team together. When they send out the call, then you just apply. I don't know if that's advisable, or you feel like it would be a waste of time because something can change, or can you adjust accordingly? Then the other thing is, when you look at many calls nowadays, they tend to want things that are really practical and will have impact. And working with the communities, and the communities can be anything. That could be the farmers, yes, it could be industry. I don't know why in Kenya, or I would say generally in Africa, industry doesn't work closely with researchers. Actually, in other countries, it's industry that even funds some of these research, so that the researchers develop products that are necessary for the industry. But I don't understand why in our case we don't have that. Can you maybe comment on that, and how can we get to that point where industry understands that we have to work hand in hand so that we can do research for them and do research that addresses the local context? Thank you.
Okay, thank you. Let's have a third one. Yes, there's one there.
Is there a way we can be part of the bodies that were mentioned at the tail end of the presentation? Because I saw Moi somewhere there, and other organizations that at least motivate the young researchers like us here to be part of academia and the research work in our global community. So is there any way UOE can upgrade or be part of the bodies that are in there?
Thank you. Paul, maybe you can respond to those three.
Okay. So actually on the question of I'll begin with the disconnect between academic and research. And I think this comes out of just one aspect or emphasis of the institutions. Like, for example, when you describe, let's take University of Eldoret as an example. because it's a general question that cuts across the institution. So University of Eldoret, would you describe University of Eldoret as a research-intensive university? Why I'm asking that question is that wherever you hear people going to America from Africa, you see someone has gone to meet a collaborator, and all collaborators we have from America, all of them are usually based in universities. They're not based in research institutions, isn't it? Because wherever you sit here, you say we are collaborating with the University of Michigan. We are collaborating with Oxford University. But you rarely hear someone saying, we are collaborating with the Genomic Research Institute of America. It's always a university. So this takes us to what I term as what I'm terming about the disconnect, is because of where the university is putting most of the emphasis on. And I'm giving another very good example in the sense that what happens typically, I'm saying this because I've also been in the university and I've also been in research. So if someone is hired by University of Eldoret, I'm just using this as an example because it cuts across all our public universities. The emphasis is always on teaching, not on research. And that's where I say that the university hires the best of the pool, of the cream of this country. But once people come in, the university structure is skewed towards academia. Lecturers are put a lot of pressure on meeting deadlines for marking, doing all those things. But you hardly hear anything about publications. But the issue of publications and patents and so on only come out during the promotions, when people are applying for promotions. But this issue does not come up every semester, as we say, the same way it comes out when people are teaching. Because for teaching, there are very clear milestones on when and what should be done. But promotion, let me use the word promotion, is optional. Isn't it? In a public university, I can choose that I don't want a promotion, but I have to deliver on teaching. I have to deliver the marking. I have to make sure that so that I can remain on payroll. But if I don't want a promotion, there's nowhere in the contract or employment where it says that you must be promoted. If you're not promoted, then something very bad will happen. Nothing happens of that. So what I'm trying to say is that the disconnect between academia and research is because of we are more inclined towards teaching and not research. And usually a good university is one in which the professor has some research that are ongoing in that particular field and also teaching in the university. And then the professor teaches in class the research he's doing in the lab. And then he has postdocs and scientists who are helping him in teaching, because he cannot do all the teaching and the research at the same time. That's why we come up with that disconnect, where our emphasis seems to be a lot more on that's our major disconnect comes in between academia and teaching. So the other thing that you asked was about the training opportunities, isn't it? What is the question again? The training opportunities on?
How can we be able to get the information for the benefit of our students who are maybe at the tail end of their training, so they can start applying for this?
Yeah, actually for our program, like what we have here, is that we usually advertise. And we have a website which I can show. If you just go to that website, you get all that information. And additionally, we also have, I think, the same thing that the third person asked about inclusion of them in our work, is that we have bi-weekly seminars, and we have bi-weekly seminars which we showcase our work, usually presentation by people across the region and the US. The work that's coming up, and in all that, we usually show the gaps that are there in knowledge. and where they would be able to come in just to enhance this level of collaboration. So while on the same, that to answer the last person, because they're all related, is that for the university to be part of our work, we are open, we are inclusive, and our research now is focused a lot more on vector and vector-borne diseases. And if the university, and people in biology, because it's more on biology and more a lot on diseases, are interested, we can, and we are more than ready to incorporate University of Eldoret, as long as it's of interest, it's something that's of interest to them. But the most important thing is to visit our website. Donald here can give the website, the information, and as well as attending the seminars that we do on every other week. And I think he can be able to share that information with you. And it's out of that if there's a general interest, then we are open to get that one coming in. The other question was ideas. You asked a question of now that the issues of timelines, isn't it? And the question was whether you should write a proposal in advance. Because as we said, and as I've described, writing a proposal is not something that you want to do in a rush, isn't it? So that there are many ways of doing it. One way, as you said, that you can do it in a way that's generic, as you have said, and then later on you look for a donor. But the other thing that is also that, the other aspect that also comes out in all this is that always in all grants, in all calls that you have, all calls, and I mean all the way from NRF to the ones that are international, like Wellcome Trust, etc. There's always what we call a science officer. And this science officer, or scientific officer, for a better word, is that you can actually be able to contact them, and they will always tell you when the next call is coming, because it's also in their interest that people respond to the calls in good time. So they'll always, if you just contact them way in advance, they will always tell you when they will be releasing the next call. And they also, and they put you in their mailing list, so that when they release that call, you're among the people who also receive the information now, but you directly in your email, telling that this call, and they'll always give you sufficient time. Some of them will even open it a year in advance, others 6 months in advance, and others do their releases in a regular basis, where you know that every 6 months, every 1 year, this call will be coming up, and so you can actually prepare for it. So the best thing is, for any call, because you target an organization, just write to that organization. They will always somebody, and ask them, when is it likely? When are we likely? And the work of the science officer is just that, to advise you on when the call will be coming up, and so on. On the other aspect of working with the industry, the disconnect of working with the industry, that one, I had a grant, actually I have a current grant, and I finished another one, where we are working with the industrial partners. One of the reasons why we have the, as you have said, the challenge in the disconnect between industry and science is our focus. In science, or as scientists, we get absorbed. We get absorbed into science, let me call it science, research, objectives, and so on. And in my experience with working with the industrial partner, because I have 2 grants, the previous one was working on flies and so on, so I had a product which I wanted to be commercialized. And I also got another one again that we are also working together with MOOST so that it can be commercialized. And among the requirements, you must have an industrial partner. And coming of this, in this particular work, one of my industrial partner is actually a factory in Arusha, Tanzania. And one of the things that I came to learn a lot out of all this, and I'll give you a very good example, is that now when I met with the industrial partner, their focus is actually very different from the scientific. So in short, while you are focused so much on the experiment that you're doing and then you look for industrial partner, the industry will engage with you when you do it the other way around. That's what I learned. You go to the industry and you ask, what are the questions they want answered? And an industry is usually based on bottom line, money. Like, for example, if I give you an example, the product that I had was one that could be able to help farmers to protect the animals from flies. And the product I had was very, very efficient. It could be able to last for 6 months. And the one that in the product, in the market, goes for something like around 15 days or maximum 1 month. And so I went to the industrial partner, and as we're sitting down, they asked me that, oh, for how long? So how long does this thing product work? They told me this is efficient for more than 6 months. And they asked me that, oh, so that means that I will only sell 1 product, 2 products in a year. Yeah, so this is a question that, while for you for science, that is good, isn't it? But for the businessman, that's not good news. They tell you that, will I sell? They told me that if you want us to continue this work, then you have to reduce its efficacy to 1 month, so that I can be able to sell more. And they ask questions about the market. And you see, we do our research, but we don't do it more with the product in mind. You do it for the purpose of science. So to say that in short, the best thing is, once you have identified the industrial partner, because that is the key thing, industrial partner that you want to work with, go there and ask the industrial partner what the challenges are. And number 2, for you to be able to work, even if you leave the industry, as you say, the industry, and you give a very good example of working with the community and so on and so forth, go to the community and understand what their problems are. Because the challenge you have always had as a scientist is going out there to addressing a problem that is not a problem. Like, for example, I was involved in mosquito net program. I think you people have heard of this, by donors, and we gave to the fishermen in the Lake Victoria region to reduce the malaria transmission, etc. And then when you went back, we find that they're using they're using the mosquito nets for fishing. So that tells you that actually their problem is nutrition, isn't it? Yeah, their problem is nutrition and not malaria. So maybe you need to address the issue of nutrition first before you can start talking about malaria. And so that means that now, in terms of research, if you are to work with that industry, let's consider it as an industry of a community, then you have to package. While your work is on malaria, you have to package your product together with how to get fishing nets. Yeah, so you have to go back and try to look at the industry who can provide more efficient, and that's the kind of grant you now write to the donor. You say, well, our work is to go there and solve the issue of the net, but we're also putting a budget here for fishing nets. Because if we don't give on the fishing nets, these nets will not find utility. Yeah, so again, the idea here is always go back. And I remember the very first time when I did my first grant with the industry, and the donor asked me that, have you, when we said, every time you would sit down and say, we're improving on our product to make it better and better and better and better. And they asked a simple question that, have you asked the farmers whether they want it? Yeah, and they say, unless you ask the people, the end users, whether what you're doing is relevant and of value to them, in science you get lost. And that is where, to answer your question, that's where the disconnect come in. You're doing a lot of research that is basic, while industry is interested in applied, what will solve a problem. And that creates in the disconnect. But if you go to the industry and you say, I'm working on this and this, before, not after, you do the work before, and you say, I'm working on this, and these are my preliminary data, how would it be useful to you? Then they will tell you what is really important to them. Then you come back and you develop your product, and then together you move together. Yeah.
Thank you for those responses. Do you have any other question coming from the floor? We've got one from an online participant. The question is, a case where the funder or the call requires the institution to support 20% of the budget, how can this be addressed if the institution is not able to cover the 20%? Is there usually a window to negotiate with the funder and at what stage in proposal development? So I don't know if you can respond to that.
Yeah, that's a very good question, and I've had that experience. In the last 2 grants that I've had with the CEDA and BioInnovate, one of the requirements was that the institution must give what they call matching funds. And the matching funds they considered here was around 20% matching fund. Initially it was 25% matching funds. So essentially we got this grant for $1 million, and the institution was supposed to raise $250,000. And then the donor provides $750,000. So the way we did it and how many donors do it is that whatever donors request for this, they're not usually requesting for the money in a check, something you can take to the bank and say upfront. What they usually require is costing of some of the, you remember we talked about environment. So you can come in and cost all those aspects. And another thing that people also do is that you can also throw in your personnel salaries. Because in this particular project, when someone is working on the project, these people are employees of the university. And the university will be paying them salaries anyway at the same time. So you can throw in, and then you can also cost the infrastructure per square meter, the rooms, the offices, the vehicles, the furniture, and everything that will be provided. And that one you can cost it and say that that is the institutional contribution. That is how it's addressed. So it is never about putting together $250,000 and presenting it to the institution there. So they ask, in essence, they're usually just asking you, how much is the institution ready to contribute in kind, or directly or in kind? Yep.
Okay, thank you. I think that one is adequately addressed. If we don't have any more questions coming from the floor, then I would like to invite Professor Gela Simiu, who's the chairman of the board, to come, and I think he knows what he's meant to do, and then we'll proceed from there. Thank you.
Colleagues, good morning. I think let us appreciate the presenter. Thank you very much, Paul, for your inciting presentation. I would like to request that you come over so that after this you will have something to remember University of Eldoret about. And DVC as Professor Beatrice kindly come forward so that you help us present this token. Slightly move so that she's here. Yeah. Prof. Closer. Thank you. Thank you very much.
Thank you so much for this opportunity.
Thanks.
Asante sana.
You are welcome. Take your seat.
Thank you.
Yeah. Colleagues, I would like to announce on behalf of the Secretariat and on behalf of the Directorate. Recall yesterday we said we shall honor this day because it's a holiday, an environment holiday. So from here, we request that we go and join the rest of the nation to plant a tree. And for us, the University of Eldoret colleagues, we shall ensure that the tree will grow. And for the visitors, we shall take that responsibility on your behalf. You will plant, but we shall make sure it grows. And anytime you come around, please remember where you planted the tree so that you check on it. After that, we are going to have tea. I hope it will be ready. Then we come back because there is a presentation online. So we shall be ready for that presentation which is online. So colleagues, from here, let us be guided. We go plant a tree, and then we come back for tea, and we come back to the hall for online presentation. Thank you very much. Asante. Karibu.
and gentlemen, let us make I don't know, a curve so that we can take a group photo. And then from there,
we na kijiko tafadhali tusonga hivi kando.
Mnatuangalia kwanza. Na mnatuangalia.
Wifi mkisimama hapo mnazuia kamera. Tunasonga. So whoever is planting nobody should wait. Jana wa kijiko.
But how do I fill the hole with Mara? I'm sure.
there's also the other side.
The group will take care of itself. Don't worry about Mara. Mara does not support growth. In fact, it will help the water to get full of nutrients.
Prof, look at me. Kidogo kishika hiyo. I want to see your face. Thank you. So anyone mwenye ako na
long press. Welcome, welcome, welcome. Those who are still walking, I'm welcoming you to sit down so that we can start. Are you indicating we are starting with online? Is the person ready? Okay. I'm waiting for the rapporteur to come over, come close. Ama unataka kufanya huko nyuma? Thank you. You are sure they can hear as we prepare? Okay. I believe we are ready, and I want to welcome you again once more for these presentations that are going to take place onwards. Some may be physical, some may be online, but you're welcome. Please, you can project the first presentation. Daktari, we welcome you to give us your presentation. We are giving you about 15 minutes, then we'll give time for questions. Please go ahead.
Can you hear me now? Hello?
Yes,
Am I audible?
We can hear you. You are very audible. Go ahead, please.
Oh, thank you. So, I'm Chibole and I'm presenting a paper on, I've linked environmental security, water security, and food security. Why am I linking this in Chepkwele-Sergoit water catchment? This because I want to promote sustainable management of Chepkwele-Sergoit water catchment. And let me recognize my able team. I have Dr. Daniel, who is an expert in soil, Ochakun is a geologist, Moasi deals with ecology, and Bode with food. So what did we do? We went about this by first subdividing the catchment into three depending on the topography and the land use and land cover activities. This basically find out what water, which is the key ingredient here, the storage capacity. And of course, when you talk about water, we had to address the source of water, which is rain. So briefly, we tailored our rainfall circle, water circle, to suit our catchment. And we did this by having the catchment subdivided into three zones. As you can see, here's the minute. This is the catchment. Now, the most important thing here is to find out the storage capacity in the catchment when it rains, how much water is actually stored and how much is lost through runoffs evaporation. And to do that, we had to look into the geology, then the soil structures, and then the hydrology. So you can see this is our catchment. The three zones, the upper, lower, and in between the middle. So these are the catchment characteristics. And now this is, we're comparing the data, how the catchment was 25 years back. That's 1995 and 2020. The impact has been really considerable if you consider the vegetation, particularly the forestation zone. Like if you see the upper catchment, most of it was forested. In 1995, right now, it just spread sparsely. Amount of forest trees you can find. Then, because of the population pressure between 1995 and 2020, you find actually the prior forested areas have been cleared, and the prior agricultural areas actually have been turned into built environment. Now, if you go straight and look into the rainfall variability, the catchment between 1995 finds, graph shows, there's been a tendency towards varying, reducing in both the amount and storage capacity, as this other graph shows. You have, if you consider the evaporation, evapotranspiration to the rainfall runoff, you see the trend, the ratio also getting less and less between those 2 decades. Then here now we have the geological map. The geological map, helps us actually find out the areas that can accommodate, are able to recharge the underground waters. You find the upper catchment and middle catchment are generally the common geology is the upper Ushangishu phonolites, which is underlain by massive, actually, basement rocks. These ones are impervious. So you find tendency of underground water storage is minimal here. The lower catchment is, predominantly the lower Shangishu phonolites, but they've been fractured and weathered, so there's room for accommodation of water. But the disadvantage is that, most aquifers there are unconfined, so the storage of water with time, after the rains have gone, if there's massive obstruction, some of them tend to dry up. Now, here's an indication of how, you find the underground water storage. You find that most of it is in the middle and the lower zone. Now, soils also help in infiltration of water after the rains. And you find that in the upper catchment, which is rich in the humic nitrosis, actually there's a tendency. You have those soils are as thick as 1.5 meters, so there's a tendency for them to allow infiltration. And this is the key area. After the rains, this water actually recharges the Chikwelel Sergoet River. This used to work very well when it was completely forested, but now with the clearing of forests, there's a tendency now to erode the soil during rainy seasons, given the topography. You have what you might call something like 15% rise in the gradient as you move from the upper to the lower catchment. Now, overall, these years you find that the amount of surface area in the river, rather the amount of water in the river has tended, to decrease. The exceedance probability of getting over 6 cubic meters per second is actually less than 10%. So with time, we're getting less and less water in the river. So we also looked at the water quality and found that in the upper catchment, we're basing that on dissolved oxygen. The upper catchment is fairly well rated, and the water quality index is much poorer when it comes to the lower catchment. The ecosystem health more or less follows the trend with the water quality. So it's worst in the lower catchment, but because that's the area that it's more populated and it has been cleared, you find that forest and vegetations are at minimum. So in conclusion, unless we take drastic measures to reserve those areas that actually recharge both the rivers during dry season and the aquifers, then this is going to affect our food security, because again, we discovered that most cases prior, where you used to have large-scale farmers, lands have been subdivided into small plots, small acres, and most of these farmers, times during dry season, when they need the water most, and the water is not valuable, they actually tend to do some irrigation. So this link, if we want sustainable management, actually, we go to identify the key areas that need to be protected. Otherwise, thank you, and also thank the University of Eldoret for having provided the grant for this study. Thank you. I'll welcome any questions.
A presenter, please. Thank you, Bwana Chibole. I want to request if there are any questions, quick ones, maybe one or two. Yes, Prof.
Thank you, Chibole, for your good presentation. I just have a question. I don't know if this is something you looked into. Because you're looking at different aspects along the profile of the Chepkoilel Segoit catchment. You looked at the soils, you looked at, I can't remember the other aspect.
The geology.
The geology, yeah.
And the ecology.
And ecology.
Then the hydrology.
Yeah, so did you find out whether there was a change in profile of the vegetation, considering the different aspects that were changing as you moved from one point to the other? Thank you.
Yeah, it's a good question, and it's all captured here. If you look at that map, land use, land cover, between 1995 and 2020, generally, actually, you find that as you move from the upper catchment to the lower catchment, the population density increases. And of course, with the people, they need to get things to, things like fuel, things like building, things like so upper catchment. If you look at this picture, you see the green area, that was the forested area before, and this is how it looks like in 2020. And if you look at the population density, those red dots, you see all these areas now. Before they were sparsely populated, and now they are really densely populated. So that has, of course, interfered with the nature of vegetation and even the hydrology of the catchment. I hope that answers your question.
I was just going to ask, he's nodding, which means he's satisfied. One more opportunity if there is. I can see there is nobody raising their hands. So Bwana Chibole, we thank you so much, and we appreciate your presentations. Let's give him another round of applause, please. Thank you very much.
Thanks to, yeah. Thank you.
Thank you.
Most appreciated, yeah.
You're welcome. We are going to go to the next presenter, and this is going to be a physical one. Then after the physical one, we'll resume the virtual ones. The keynote speaker indicated that there was something that he wanted to share with us, and let's give him this opportunity, please. Thank you. Thank you.
I'm just going to, I'm going to give you a small highlight on some of the research that's going on in my lab in KALRO, and as well as potential areas of collaborations that can come up between us and the institution and University of Eldoret. So I start by actually just indicating the number of institutions that we have collaborated, that I collaborate with and have collaborated with in the work that I do in the lab, right from KALRO, Kentech, and a number of institutions that I've as indicated down there in the work that I do here, with funding from NIH and by Innovate Africa, Grand Challenges Canada, etc. So this is the team. This is what makes up the people in my lab. I have Hassan Ali. He's now retired, but still gives a lot of background support. Rosemary and Winnie, they do a lot of the work on molecular genetics in my lab. Kennedy is a vet, so he does for me, runs most of the chemical ecology, the veterinary aspect of the work. And Abraham Ayoke now in Congo, a little bit of population genetics. Benson is a chemist, so he runs most of the chemical analysis. Bilia, she's now just finished her master's, going for a PhD, but her work is more on bioinformatics. And then Clarence runs most of the molecular work on mosquitoes, or mosquito work that I'm going to present. So I'll give a little bit, very quickly, give you some highlights on the work I work on, mostly on crescent trypanosomiasis, research and development, and I also do some work on mosquitoes and parasite research. And then I'll also talk about the vision or continued collaboration that we can have with the university in a way that anyone interested can then reach out to me. So my flagship program is actually based a lot on tsetse trypanosomiasis. And as you can see there, we have the tsetse fly colony, and they don't lay eggs, they basically give birth to larvae, and they also feed exclusively on blood. So we have our One Health focus. We have trained several PhD and postdocs, and we have 2 patents. We have over 60 patents peer-reviewed, certified by the students alone, and significant amount of money, and we have over 10 years of support from NIH, and we have several North-South collaborations, and we have been able to pull together close to $10 million. So one work that we are working on tsetse trypanosomiasis is about searching for new repellents and attractants. Most of this work was done by Hassan Ali and Saini. These are ICP scientists when I was still working with ICP. And it revolves around this animal we called bushback. Yeah, not bushback, but waterback. So they discovered that tsetse flies tend to avoid waterback. And with that one, they look at the profile of the waterback, at the chemical profile against the buffalo and oxen, which the animals, the tsetse flies like, and then identified this column, this team around here that seems to define waterback. And they characterized it and found that it has 15 compounds using a chemical analysis. And out of this, they identified that actually one of these compounds, a ketone, fatty acid, phenolic, and delta and lactone, will make an optimized repellent, which when they did the experiment in the field, they found that actually these ones are providing 85% reduction in feeding efficiency of the tsetse flies. So that is a good product, and subsequently made it as a product that could then be used to keep the animals, tsetse flies, away from the animals. So Hassan Ali and I picked it up from there. And among the things that we came here is that we took this molecule that we are referring to here as delta-octalactone. And when we looked at it, and we took the analogs of it, and when we tested the new analogs, we found that actually these analogs, one of them was a more potent repellent than original parent molecule, and another one had more of a repellent property, more other attractant properties. And when we look at the one that had attractant properties, we formulated it together and found that the attraction property was actually was similarly as attractive as the animal itself. And so we felt that this could probably be a new attractant for the tsetse flies and something that we are working on. And while the repellent also seems to have a better repellence than the original molecule, and when we did a benchmark analysis against the original compound, here we find that the original repellents, our repellents was actually twice as effective as the original molecule, original repellent blend. So we have these 2 patented, and we also have this one. And then from here, we came out in the field to see how effective are these 2 molecules, our new repellent. And as you can see here, on one side we have the repellent animal without the repellent, the white one without the repellent, and the black one with the repellent. And you can see that where the tsetse flies, and you can indeed see that this one is clearly agitated. There are many flies flying around, while the one with the repellent seems to be quite effective. So we felt that this is something, this seems to be like a very effective novel repellent that we could probably take out there, to the market and see whether it can have some economic value. So as you can see on the left, the animal is wagging the tail, trying to push away the tsetse flies. And then we went in and looked at what you see at the screens down there are sticky panels that tsetse flies attached to. So a way of sampling the population of tsetse flies around. And so when we looked at the flies that were caught in those panels, we found that there was a suppression of flies by over 90 different species of tsetse flies. And for Pallidipes, which is one species and another one, Osenai, we found that there was a very drastic reduction of flies. The first column here shows the number of flies that come in without any control for the oxygen, the white oxygen, and then this one shows the number of flies that come in with the trap. So you can see the drastic reduction by almost 95%. And so when we look at and this is another species that usually there's no repellent has been described for it, and for the first time we're able to show that our repellent is effective against Ostenai. And so we had a complete reduction by where we had 82 to 0%, while the new one, there was a reduction of feeding efficiency from 82% I showed earlier to 0%. So that's an effective repellent. But when you look at this one against a species that we find in Western Kenya, in Western Kenya around Lake Victoria and in riparian regions in Uganda, both repellents are not effective. I'm suggesting that there's a need to do more research in that area, and that's what we are working on. So this repellent was actually effective against 5 species, suboptimally effective against 1, and not effective against the one that I'm just showing here. We call it Glossina fascipes. It's found along Lake Victoria. So we did this work in Tanzania for the Submorsitans centralis, in Uganda, Kenya, Uganda, and Tanzania. So we decided to add some more value to this product, and by adding value to this product, we decided to encapsulate it in beta-cyclodextrin, and we successfully encapsulated in beta-cyclodextrin such that when you look at it against non-order that is not encapsulated for control release, we found that actually that delivery system was more effective and also used biodegradable non-woven fabrics. And these woven fabrics, we worked with them, we made them into rolls, and in these particular rolls that can be used to make fabric and clothes. So you can have clothes that when you put on, you repel most of the vectors or that can spread diseases. And we also found that the smell of our repellent was smelling more like a buffalo, like a waterbuck. And so we worked with a French company, and we were able to mask the smell so that it smells like a perfume or a cologne. So we went back into the field and we tried to see how long does this last on a cloth. If you make a cloth, because you're looking at people in the military security services who can actually use it when they go out to the other rangers and so on, and even tourists. And so we find that when we went there, when you put this one on the cloth, it's a trap of flies with and without this one repellent, without the encapsulation. And we found that actually the repellent, when you put it was quite similar to without the encapsulation. And so we saw that having this as a new product would be a good business line. And so we put them out in the sun, and we found that the sun actually has significantly reduces the efficacy by looking at the product here, because the attraction of the fly increases despite the repellents. But when exposed for 30 days in the shade, the reduction is still maintained for some time. So this one was better than this one, but this we're just trying to see how would this one be able to survive in the presence of sunlight. And so we have enlisted the work of our industrial partner, and now we are now going through what is called the Valley of Death. This is a path that is normally taken by all products development. So we are now at the point of technology transfer, and we are looking at aspect of commercialization, working with the industrial partner. So this is usually the path that's taken by all products, and we are all trying to get out of this valley of death. And so in progress, we're doing safety trials on animals, pour-ons within, and doing pour-ons within integrated vector management, and improving the repellents, and then trying to work on whether it can also be effective against other vectors like ticks. So here is a cartoon that shows how flies locate their hosts. They usually fly from a long distance by smelling molecules that are very light. This is carbon dioxide, octanol, and acetone. These are things that animals breathe. And so when they breathe, they go very far. And so the fly can pick it up from very far and locate where it is coming from. As it gets closer, then it gets closer to where we have the animal. The smell of the urine, this comes from the urine of the animal, and then it can be able to locate the animal physically by looking at it. So this information was actually used by my colleagues earlier in Zimbabwe, and and also in Kenya to make a molecule that was called poker that is used routinely for commercialization. So we looked at our repellent here and our new attractant and trying to see how does it compare with this other one and also with the animals. So we did the same experiment I showed with the field, with the different animals in the middle, and with that we could be able to see how many flies are coming in, and we found that our repellent here, our attractant here is just as effective as the animal in attracting the fly. So that means that it has a very strong pull system, so it can pull the flies pretty much away from the animal if we kept at a spatially located area. And we find again this is very effective against all the three except auceni and against fastkeepers, our attractant. So this is on work that we are coming on. And so the two of them, attractant, the new attractant, new repellent, can be integrated in what we call a push-pull system. So we are trying to identify the new repellent attractants for this particular fastkeepers. Here we have been trying to see if we can be able to get identify new hosts that the sesefly feed on along the coast, and also trying to see if we can be able to get new repellent from the monitor lizard, which the fly has been found to feed on, and also from other sources of like the oil formulations. And then we have also been working closely in identifying with our colleagues, basically testing for our colleagues some of the molecules that have been identified, like this is a new identification of a new pheromone from the fly by our colleagues in the US, and so we have been trying to work out and see how this one works out in the field using the different physiological tools. So we have also worked along and trying to look at the molecular basis of olfaction. This was driven by initial study that we did on annotating the genome of the sesefly. And this was work that was funded by the US government, and it was a community initiative. So we did this work and we identified, we sequenced the genome, and my role of my lab in that work was to annotate the genes that are associated with the sensory aspects, especially the olfaction aspect of the sesefly. So we were able to identify the receptors, and we are able to compare how these receptors compare across the different species of seseflies and the 6 of them. But the goal of doing all that was to be able to identify what makes the differences between, as I showed earlier, the sesefly are responding to some, our repellent, and others were not. So we were trying to see, can we be able to look at a molecular basis of doing this? So we went further and looked at whether we could be able to see whether there's an expansion of some genes, specific genes that are associated with one species and not with the other one. I employed a little bit of bioinformatics approaches to see whether we could be able to use those tools to identify this new method that genes that could be able to decipher these different molecules. And then finally, we started working on filling the gaps that have actually been identified in this one using molecular approaches, and then characterize the sensory system that actually used in that work, and working with our colleagues. Our colleagues actually identified their molecular basis. So among the things that we are looking at here is a drastic reduction on some of the genes that are involved in olfaction in seseflies. And our colleagues back then in the US were able to actually identify some of the sensilla and the expression profile of some of the genes that are associated with the responses to these particular molecules, and trying to see whether we could be able to use these responses to identify new attractants and new repellents. Like, for example, we find that the sesefly is actually responding to 2-pentanol that had not been identified before, based on the Drosophila model, in which this Drosophila model is where they took the sesefly gene and then it is actually expressed in Drosophila, and trying to see how Drosophila works, and then we're able to identify pentanol as a new molecule that we are now testing out in the field. And on molecular genetics work, we have been trying to see whether we can be able to look at connectivity between the different sesefly populations in Kenya, to see whether the flies actually move between one area and another. And this was driven by realization that ability to attempt to control seseflies in Lambwe Valley has failed. Despite reducing the fly population down to undetectable level, the fly population always resurges. And the question was whether that population comes out from the same population that is growing again, or whether it's an invasion by a population that is coming out from outside, and also trying to identify which populations in Kenya are actually isolated, so that in the process of eradication, if they're eradicated, the fly population cannot be taken in again. So we have this work that's been going on by Rosemary and Winnie, leading the show on temporal differentiation, phylogenetic analysis of this fly population, as well as in southern. And then we have also had this work going on in Congo-Brazzaville, where we have description, trying to have a better understanding of which fly populations exist there and the migration patterns, and as well as trying to employ AI, machine learning approach in integrating genetic ecological aspects. And this was more of coming up with more of a predictive model that can be able to help in development of an atlas of fly populations where they can be identified using remote sensing and GIS. On parasite-host interactions, we have also tried to decipher, again, like in olfaction, why some flies are better vectors of trypanosomes than others. For example, Pallidipes here is more resistant, and the overall goal being to be able to overall come up with a transgenic fly that does not transmit trypanosomes. So you have looked at we have characterized the trypanosome, we have characterized the expression profile in the midgut and in the proboscis using our PhD students, as well as trying to look at the genetic aspect of the trypanosomes in relation to the regions in Kenya, to try to understand how these relationships works out together. And looking at also on how some of the parasites are better transmitted between different hosts at the proteomic level and so on. And the gut microbiome. There's a lot of work that has taken out of interest for us now in the microbiome, because there's now an understanding that most of the parasites are driven a lot by endosymbionts. And so there are some endosymbionts that have recently been identified that actually enhance or reduce vector competency in the fly. And so we're trying to see, can we be able to enhance that aspect in the fly so that we can have fly that do not transmit trypanosomiasis. And then we have a work going on reproductive biology of the fly. And the goal here is to trying to see whether we can be able to come up with abortifacient compounds that can actually be used to make the flies abort before they give birth to the larvae. So this is work that's also going on. On mosquitoes, our work has focused a lot on trying to understand why do we have why do you have mosquitoes breeding in polluted habitats, and yet we know the malaria mosquitoes should not be doing so. So we look at the flies mosquitoes in Malindi and Kisumu, and we found them in dirty water or polluted water, which was strange. So we came and looked at the biological cost, and we found this comes at a significant biological cost to the mosquito, in the sense that if you release that pressure, the population will be replaced. But if you maintain the pressure, this strain will be retained and continues the flies moving on. We have done several studies, again, trying to see, number one, what are the molecular bases that support those transitions, and also trying to understand if this one would also be related to insecticide resistance in the mosquito by using the cytochrome P6, P450, which is actually used to metabolize insecticide. So we have also gone back into the same mosquitoes and look at Moraya conegi. This is a curry plant actually to see whether it can be able to repel mosquitoes. So from it, we have been able to, we found a bioactive molecule that when you put in the water, it can actually kill the mosquitoes. And then we have looked at the molecular basis of this response using transcriptomic analysis. And we have also been looking at whether this can be used as a fumigant in a place so that you can be able to, a way of reducing mosquitoes, and looking at other aloe as a plant, whether it also has those potentials in larvicidal activity against the mosquitoes. So other work that we've done here have revolved a lot around mosquito work, Plasmodium mosquito work, looking at the microevolution, changes in genotypes. For example, trying to understand whether we're trying to see whether chloroquine should be reintroduced in Kenya. We found it shouldn't, and using different methods of identifying different species and flies in the mean parasites in our system. So we've also been doing some work on aspect of entomology, but this is more contributing mainly on the approach and analytical work on ceratitis and Schistosoca gregaria, which is actually locust. So we trained, these are the students we have trained in our program, around for the PhDs, and as well as we also provide master's training program for London School of Tropical Medicine in Nigeria. Every year we provide this training. These are our new postdocs, as I mentioned earlier, that we have recruited into the program. And this is the team that we have been working with in our training. So this is the grant support that we have received, but these are the currently ongoing work in our program. And these are actually the collaborators we have worked with in bringing us up to where we are. So in collaboration, we continue One Health, we build on innovation, transformation, and considered collaborators where they are, so that we can have a long-term investment, and so we can co-implement any work that we have, take stock of what we have under both teams, and we can document work that's going on, and so on. So long-term, we do basic research from what you have seen. We also do a little bit of applied research, and we're also expanding our portfolio to other arthropods. Anyone who have an interest in this one, and then we also do our training program that I mentioned earlier in the day. And this brings me to the end. Thank you so much. Thank you so much for your time, and thank you for listening to what we have.
Let's do it better. Let's do it better. Thank you. Thank you. I'm glad that it was spontaneous. I didn't have to ask you. At this point, I want to find out if there's anybody that would like to find something. I think, Prof, your time.
Thank you, Paul, for your presentation. In your field experiment, I noticed you allowed sampling error because you had color coming in, a white animal and a black one. And you mentioned that sesefly is like buffaloes. Of course, the black one is similar to a buffalo. And you also mentioned that as they approach using scent, they use fisco when they reach closer to the animal. Don't you think there is an extraneous error there?
Thank you, Prof. Let's see if there's another one. We'll take two, just two. I'm glad there's none. Please respond. White against black before I ask mine. Thank you.
Yeah, so let me go there again. Very quickly. I want to show it here. So this is a bit, and yes. So I agree with you that seseflies tend to be attracted more to black than to white, which is true. And that's why we put the targets for controlling sesefly as black targets and not white. And also for traps are also made up of some black material. So naturally, given the situation that we have here, if the black one, if the one that is swelling was white, you would actually expect the flies to avoid it and actually go more for the one with the repellent, but it's black. So this just goes on again to just reinforce what we are saying, that our repellent is quite effective, even despite the fly that, the animal that was black in color, having a repellent, it still could not attract the flies. So yes, so normally, this is what I've shown you is representational of the animals we used, but sometimes in nature, it's very hard to find an equivalent animal to compare with because we are now working with animals coming from farmers. So you are right. Yes, the flies come in, and so that is something like a caveat. But nevertheless, we expected them now to be more attracted to the black one anyway, but it didn't. again, talking about the veracity of our repellent.
Mine is a short one.
Okay.
I saw a large number of collaborators.
Yeah.
Are they all current or some are now out of the picture? Thank you.
Yeah, the number of some of them are out because our collaboration is something that's dynamic, but most of them are ones that we are still working with, and even some that we are working with are not included here, are not included in this list. So this is not an entire group of collaborators, but collaborations is never stops. So it's usually a continuous process, moving from project to project.
Round of applause, please. Round of applause. We are going back online, and I'm requesting if Langat is there. Are you there, Bwana Langat? Thank you very much. Ayuko?
Good afternoon, everyone.
Good afternoon. We are going to ensure that you keep within the time that is provided for, 15 minutes. If you go beyond that, you'll hear my voice coming through. So
Okay.
try to keep within time, please.
Okay,
Starting
right,
now.
Thank you. I'm Mr. Philemon Langat, and we are working with Professor Jenga, Dr. Pascaline Cheruto, and Professor Lukaba from the University of Nairobi. The title of our presentation is Molecular Phylogeny of the Kenyan populations of Dodonia from the North Rift and the coast using ITS1 and RCBL gene sequences. I'll give a brief introduction. We are working with this family Sapindesi, which has over 1,900 species distributed in 142 genera. The genus is comprised of over 70 species. This plant, Dodonia viscosa, is of Australian origin with 7 subspecies. And we have 3 of them outside Australia, and only 2 varieties are found in Kenya. The plant is used medicinally around the world, and it is a plant that is of has some taxonomic difficulty because it is regarded by some taxonomists as a subspecies. Some call it regarded as varieties, and some even as a species. And because of that, we were able to perform an in-depth taxonomic study to delimit the 2 varieties based on their DNA sequences. Our study area, we collected the plants from 4 counties: Baringo, Elgeyo Marakwet, Uasin Gishu, and Kwale. Those are the 4 counties, and from that we were able to extract their DNA using a sit-up method. After extraction, we did gel electrophoresis to confirm the presence of DNA, and we were able to use the primers ITS1 and RBCL for amplification, that is for amplification. This chart here shows us the primers, specifically the sequences, both the forward and the reverse sequences that we were able to use. After extracting the DNA, we took them to Macrogen Europe for purification and the sequencing. And we obtained the results in 3 forms: AB1 file, the PDF, and also as a text document. And the results also were inclusive of the forward and the reverse sequences. Now, from the sequences, we were able to align them using a software referred to as Mega 11 by Muscle. And then we were able to export the aligned sequences as a mega file. And from that, we use the sequences to estimate their phylogenetic relationships by maximum parsimony. Also, we also did a blast search so as to locate regions of local similarity between our sequences
from the different populations and the gene sequences that are found in the gene bank in the NCBI database. Now, our results are as follows. The first figure represents an image from the gel imager. You can see the presence of the white spots telling us of the presence of the DNA from all the populations. We were looking at 11 populations. The next figure shows us the sequences that were obtained after alignment. You can see there are some gaps in the sequences. You can see there are some mismatches, and even most of them are matching. From those sequences, from the ITS sequences, from the forward sequences, we were able to get a total of 3152 nucleotides. 381 of them were conserved, 2505 were variable, 716 were singles, and 1672 were parsimony informative sites. This is a third figure showing us the results from the ITS1 reverse sequences, and still we are getting similar results. You can see the matches, the mismatches, and the matches. From the reverse sequences, there were a total of 775 nucleotides. 256 of them were conserved, 511 variables, 210 singletons, and 285 of them were parsimony informative sites. From the RBCL forward sequences, this is what we are getting. From the alignments, you can also see there were some gaps, there are some matches, and even most of them were actually matching as a result of the alignment. From that sequence, we can see there are a total of 1729 sequences in total. 864 of them were conserved, 897 of them were variable sites, 685 of them were single sites, and 207 were parsimony informative sites. From the reverse sequences of the RBCL, we're able to get this. This is just a section of it. You can still see there were some gaps, there were some matches and mismatches. And from those sequences, we had a total of 1767 nucleotide sequences. 694 of them were conserved, 870 of them were variable sites, 479 of them were singleton sites, and 63 of them were parsimony informative sites. From those sequences, we were able to develop phylogenetic trees to show the relationships between the different populations of Dodonia viscosa. And if you can see clearly, the gassy populations are the ones from Kwale. You can see there are 3 of them, the gassy populations. And we are trying to compare the populations along the coast and the ones which are upland. And we can see from this that there is no disparity amongst all the populations. If we look at the river sequences, the ITS if you look at the RBCL river sequences, you can see the populations along the coast were demarcated from all the others. The next phylogenetic tree is the one for ITS, forward sequences. You can still see the gassy populations, which are the coastal populations. they are still found within the others, which tells us that there is nothing so much that separates them from the other populations based on their sequences. The ITS1 reverse sequences was also very unique, just like the RBCL reverse sequence, which brought together the coastal populations from the rest. You can see the Gasi populations, these are the coastal populations, they're demarcated from the upland populations. Now, from the blast search that we did, all the sequences, all the 11 sequences were identified by blast as Dodonia viscosa with percentage similarities of from 94.55 to 99. And from the same sequences, we were able to identify Sideran Clofera minor as the closest relative of Dodonia viscosa. So our blast search was telling us that these are Dodonia viscosa and not any other species. We also did a genetic distance. We were estimating the genetic distance amongst the different populations. And you can see the distances, they were ranging from 0.4273 to 1.1513 from the forward sequences, and 0.020 to 0.620 from the ITS1 reverse sequences.
Can you hear me?
And also,
can you hear me?
A genetic distance of 0.00 to 0.04831 from the forward sequences of the RBCL.
Can you hear me?
0.07. Sorry?
Can you hear me?
Yes, yes.
The slides are not moving with us. We are stuck.
Oh, sorry.
We are stuck on phylogenetic trees, RBCLF.
Oh, sorry.
Number 23.
RBCL.
It is slide number 23. Can you go back to the presentation mode?
A presentation mode, all right. Are you there?
Yeah, it is moving. It is moving. No, but it's still stuck.
All right.
Go to 24. Yeah. Now you can continue.
Okay. We were comparing the sequences from the donor populations from the coast and those of the uplands. And we can see here that the ones from the coast, they are very unique. They are demarcated into one group compared to the others. This is from the RBCL reverse sequences. If you look at the ITS sequences, you can see the populations from the coast, which are the GASI 1C. You can see the GASI M5 and the GASI 3B. They are found within the other populations, which tells us that there is nothing, they are not different so much from the rest based on their sequences. It tells us that they're very close relatives. If we look at the ITS1 reverse sequences, the populations from the coast are demarcated from the others. You can see they have been placed into one group, though they are closely related to the populations from Iten and those from Sergoit. They share one apomorphy. You can see they share this apomorphy here. From that, we did a blast search so that we were doing a blast search of our sequences, and our blast search was able to identify all the sequences as Dodonia viscosa with percentage similarities of 94 to 99%. And from our blast search, We are also able to identify this other plant called Sinoraldcofera minor as a very close relative of the Dodonia viscosa, that is from the blast touch. That was the closest relative. We also did an estimation of the genetic distance using the program called MEGA, and the different populations, their distances were ranging from 0.4273 to 1.1513 from data from the forward sequences of ITS1, and also a distance of 0.02 to 0.621 from the ITS1 reverse sequences. From the RBCL reverse sequences and the forward sequences, we're also getting this genetic distance of 0.00 to 0.4831 and 0.07 to 0.433. These are the genetic distances between the different populations. Now, what we can say is that there is a very close similarity between all the Dodonia populations. Number two, we are saying that the reverse sequences has clearly demarcated the coastal populations from the inland populations, that is from the reverse sequences of RBCL and ITS1 genes. Now, from the bootstrap values, we can also say that there's a strong relationship amongst the coastal populations, especially GASI M5 and GASI 1C, which were giving us 95% confidence levels. And the closest relatives to the GASI populations, which are the coastal populations, are the ones from E10 and Sergoid, and these are evident from the phylogenetic trees. The small values which are obtained from estimation of genetic distance are implying that there's a very minimal variation and a very close relationship amongst all the Dodonia populations. And possibly, this could be attributed to the minimal genetic distance could be attributed to differences in altitude. Number two, there could be attributed to differences in the surroundings, differences in the climatic factors, the likes of the warm index, the cold index, the mean annual rainfall, and even the yearly temperature, and others. Now, conclusion, we are saying that all the populations of Dodonia in Kenya should be treated as one species, that is Dodonia viscosa, and not as independent species, as some literature is suggesting. And number two, we are recommending more research targeting other gene regions with the use of different primers, so as to clearly get a delimitation of this Dodonia viscosa. Let me acknowledge the University of Eldoret, the University of Nairobi, also Mirej in Europe, where we were doing our sequencing, my supervisors, and the technical staff from the different labs. Many thanks for listening to me. Thank you so much.
Thank you very much, Bwana Langat, for that presentation. Now, I hope you can hear us. I want to see if there's anybody with a question regarding the presentation. Yes, Prof.
Philemon, thank you for your presentation.
Yes.
I've got 3 questions. Now, one, I would want to find out whether you rooted your trees using the the close relative of for the group. I think you called it sign or something. Because the figures that you've presented, to me they appear like unrooted trees. But if you root the trees, probably you'd get, using the closest relative, you'd be able to get maybe a different topology of the phylogenetic trees. So the question is, did you root your trees when you're doing the analysis using the closest relative? Then number two, maybe it would have been good for you to do a combined analysis of the two genes, of the sequences of the two genes, and see what the outcome would be. Would it give you a different result? Did you do that? I think I would be interested to know. You know, you combine the two, the sequences of the two genes, and then do what we normally refer to as a combined analysis, and see whether you, because I believe a combined analysis would give you a more robust phylogenetic tree with very good supports for the different clades, if you did that. Then you say that the support for the coast group was at 95%. But how about at the root of the tree? If you did the rooting, was there support for that big clade? I think that would be something I would be interested to know, because if the internal clades have got good support, but the big clade that comprises all the groups doesn't have support, then that may not be a true group. So probably you could explain that as well. Thank you.
You got the three questions?
I got the first two questions.
Okay,
pay attention.
Let me repeat the third one.
Yes.
Yeah, you say that the coast group has got a very strong support, a bootstrap support of 95% confidence level.
Yes.
But I wanted to know the big clade that also comprises the coast group at the root. Does it have equally strong support? Because if it doesn't have strong support, then you might not consider it to be a real group. So what was the bootstrap support for that, for the root, the clade that comprises, that encompasses all the groups on the tree?
You got it.
Let me try to answer the first two, so that I'll find time to look for my good professor, so that I come and consult him on the same. The first question on rooting of the tree, I never did the rooting of the tree, but I think it would be good for me to do the rooting, since Professor Otieno, who is a taxonomist, has suggested so. I did not do the rooting, but it would be good if I would have done it. Number two, I also did not perform a combined analysis based on the different sequences, and still, I would also wish that I do a combined analysis so that I get a better trees based on the two primers. I'll still find time to consult with Professor Otieno, who is a molecular taxonomist, so that I consult him on more, especially on the third point, which I'm not getting very clearly. Thank you so much.
Thank you. More questions are coming, please.
Thank you, Langat, for your presentation. My question is out of curiosity. Your first experiment, you tried to find the presence of DNA.
Yes.
And even you provided results.
Yes.
And the lesson I learned yesterday is all organisms, living organisms, have DNA. So which question were you answering? What were you trying to test?
Before sending the DNA for sequencing, it's actually good to confirm that you have what is supposed to be sequenced. Otherwise, you'll be sending anything which may not be DNA. So gel electrophoresis is just a confirmatory test to confirm that what you have is actually DNA and not anything else.
Are you done?
Yes.
Thank you very much. Is there any burning one? Or we put this one in the file? Okay, let's give him another round of applause, please. Thank you very much, Manalangat, for being present and virtually and giving us that presentation. We are going to the next one. There are two more and then we'll just be done. Banachibai, are you online? Can you hear me, please?
Yes, I can hear you.
You are next. And remember, we are time pressed, so keep it short. Give us the gist of your work.
Hello?
You can start as soon as you are ready, please.
So how do I start? Hello? Hello?
I can hear many
Hello?
voices.
Hello?
Wait a bit.
I cannot share the screen because another participant is still sharing.
Just wait, just wait. It's being organized.
Okay.
Can you share, please?
Yeah, I'm trying.
Are you able to share?
Yes.
Please.
Are you able to see it?
Yes. I don't know whether it can go in a presentation mode. Oh, much better. Your time begins now. Thank you.
Okay, thank you. Okay, welcome to my presentation. My title of the presentation is Modeling Risk Factors of Miscarriage Using Pure Model. My co-authors are Dr. Akins Matiano, Dr. Ayub Banapaba, and I, John Olive Kibai. I'm a postgraduate student in the Department of Mathematics in the University of Eldoret. Welcome to my presentation. Introduction. So this discussion is all about miscarriage and modeling the risk factors that will affect it. So miscarriage is a phenomenon that is very common but not given much attention. The large number of pregnancies progress without major complications, but however, a good number will undergo adverse pregnancy outcomes.
Banachibai, can you hear me?
Yeah, I can hear you.
Can you try to put it on a slide show, please?
Only?
Slide show.
Oh,
so it becomes larger.
How can I do that?
It is supposed to be like a cup at the bottom right. I don't know whether you have it on your computer.
Computer cannot show the on the slide. Sure.
Go to slide show. Yeah. Thank you. Much better.
That one's better. It's okay. So I proceed on. So I was saying that the miscarriage is one of the adverse pregnant outcome, and it's defined as the involuntary loss, pregnancy loss at the time before the fetus can survive out of mother's womb, or a pregnancy terminated at less than 22 weeks of gestation period due to non-human factors. Miscarriage globally has the prevalence of about 10 to 15% for all clinically recognized pregnancies. Empirical estimates gives a value of 2 to 3, the lowest percentage, and the 30% highest. In Kenya, the prevalence is about 12.2%. The causes of miscarriage remains uncertain or unknown. Nevertheless, some studies have identified several factors that contribute to the miscarriage. The common cited factors include demographic factors like age, men's age, gravidity, length of pregnancy interval, and pregnancy history. Clinical or genetic factors like chromosome abnormalities and mutant genes, and some lifestyle factors like drug use, smoking, drinking, obesity, place of residence, and race. However, the risk factors that led normal pregnancy to miscarriage are fairly well known or established. In addition, determining the occurrence of miscarriage poses a significant challenge, particularly in low-income countries where there is less data on its prevalence. Hence, assessment of demographic, socioeconomic, medical, and lifestyle factors that have effect on pregnancy loss is this study is all about. In modeling the risk factors that affect miscarriage in survival analysis, mostly the model used are Cox model and acceleration failure type model. These models assume that all subjects under study will experience the event of interest so long as or provided they followed for a long period of time. But in reality, this is not the case. The event for a case which is miscarriage may not occur totally in some mothers, no matter how long they are followed. The Q model is a class of models for census survival data for subjects when some of them will not develop or experience the event of interest, so however long they are followed. So in this study, we implemented Q models to develop the risk factors that affect miscarriage and also use the model to assess the effect of these factors. Methodology. The study design was we used a secondary data. The study area was Kakamega County, Northern Kenya. The study population was all pregnant women living in Kakamega County, in Northern Kenya during the period of 1st January 2019 up to 31st October 2020. Sampling procedure, all pregnant women enrolled in antenatal care from January 1st January 2019 up to 31st October 2020 were enrolled in this study. The inclusion criteria was the pregnant woman should have enrolled in antenatal care in that hospital and have confirmed pregnancy of about gestation age of 6 to 12 weeks or earlier. The study variable here were extracted from the medical records of the hospital, and the response variable or outcome variable was time to event or time to miscarriage. The inserted variables were derived from social demographic characteristics, clinical characteristics, and lifestyle characteristics. Statistical analysis. Data exploration was done using Excel, and ARPAGate was used for estimation and analysis. Mixed Q model was used to model the risk factors. 5% level was used as the level of significance. Next, secure model. For you to use this model, you need to be able to identify that the data has a keyword fraction or has long time survivors. So one can identify whether the dataset has a keyword fraction by looking at the Kaplan-Meier survival curve. So you have to plot the curve and see if it has a long, stable tail at the end of the study. If it has that, then the population has a proportion of keyword subjects. In keyword modeling, there are two classes of keyword models that have been proposed, namely mixture and non-mixture keyword model. This study used mixture keyword model, and this model, it models the population as two groups, those that are keyword and those that are not keyword. Those are keyword means those who, well, those are long time survivors, and those are not keyword. Those are susceptible to the event. Mixed keyword model was first proposed by Tyrell and takes the form the probability alive at time t.
I want to request you move to the results, please. Results, please.
Hello?
Move to the results.
I move to?
Results.
Oh, results. Okay. So results and analysis here. So the data used secondary data, as I said earlier. The sample size was 6,075. This was from record of pregnant women from that stall from January 2019 to 31st October 2020. The response variable was time until miscarriage, and the coverage used here were from demographic, like gravidity, maternal age, and some lifestyle. Descriptive results and analysis. So table 1 indicates number and percentages of those who experienced the event and those who did not. So in that table, a total of 248 pregnant women experienced miscarriage, which was 4.1% of the sample. So table 1 display the descriptive statistic for the variables of interest. In the same table, the distribution of number of women who experience miscarriages varied across different levels of categories variables. It was indicated by the p-value below 5% if you use the Pearson chi-square test. So there's figure 1 there, which will show the overall Kaplan-Meier survival plot to show if there is a pure fraction, and the graph demonstrates a plateau beyond the threshold of 0.95. So in the same graph, it indicates that the length of follow-up period was sufficient and exists a cured fraction. So the table is there. So in that table, we have just shown the number and the percentage of those who experienced events and those who did not experience in each category of the variable. Like one of the variables was ethnicity and has 3 categories, 4 categories, mainly challenging, diffuse, and worse. Like in challenging, sample 10 got miscarriage in ensemble and 71 did not. The total were 89 for that group. In review, the total was 104, 10 got 95, and it goes like that up to the last one was parity, because we show parity was a contingent variable but it categorized. So contingent variables were categorized as shown in the table. So that's the graph, the Kaplan-Meier graph of the survival curve for miscarriage. If you plotted the survival probability against survival time, you can see at the end, so the graph does not reach 0, meaning all groups did not get the events. And control group did not get the event, some percentage and some are keyword miscarriage. So model development, the relationship between survival time and coverage was modeled using proportional hazard keyword model. We use a program in R to estimate the parameters in the model, and we started by modeling one factor at a time. The data 2 shows the results of modeling one factor at a time. We are calling it bivariate model. Bivariate here means we are modeling the response and the warning planetary variable. So in the table, the following variables: HIV, graffiti, parity, age, marital, employment status, and sexual labor application were not statistically significant at 10%. So we took 10% as the screening level where to screen factors that are significant. So those factors that were significant, they were the one passed to the next multivariate model, and there were 7 of them listed there: place of residence, employment status, ethnicity, level of education, pregnant history, and antenatal care visits. Multivariable model of these factors is shown in table 3. So this is the table 2 for one factor model. So you can see some were significant at 10% level, some not. So the table showing the parameter estimate.
Move a little faster, please.
Errors.
Move a little faster, please.
Okay. So that's the table continuation. So that's the table 3 showing the multivariable model. And among these one, 2 factors were not significant, so they were removed. And the final model is given in the table 4. So the multivariable model obtained in table 3 indicated that the college and job status do not exceed statistical significance at 5% level. So this coverage was omitted, and the final multivariable model is presented in table 4. So the fitted model is this one. So the mixture model, it has 2 parts: the keyword part and the latent part, or what we call the survival part. So the table 4 shows the final model. So those are the parameters. You can see all of them are significant with respect to keyword fraction or keyword probability. So it also shows estimate standard error and p-value for the keyword and for survival. So the keyword and their survival is what we call the latent part. So conclusion discussion is, so this paper we estimated keyword fraction of miscarriage and effect of covariates on keyword rate using models. So the keyword model shows that place of residence, ethnicity, number of previous miscarriages and stillbirth, and number of NCs was statistically significant. That is, they affect the keyword fraction. But these factors did not show much effect on the survival part or latent part, apart from the number of antenatal care visits. It's only that what affected the survival part, the survival part. So number of previous miscarriages, stillbirth, NC, and residence, their keyword probabilities were estimated and were given in the data table, given there, like 54.8% for the number of miscarriages, 47 for stillbirth, 70 for NC, and so on for the others. So this information you cannot get from the normal standard Cox model and the FT model, because those ones, they do not show the keyword fraction, but the keyword model can show this. So it shows that the factors that affect miscarriage, some are only do not affect the survival or the time for miscarriage. They only affect the proportion of those who don't miscarry. So the classical methodology lacked the ability to differentiate between curative treatments and treatments mean that only extends the lifespan. In addition, their sustained power decreases as the length of the follow-up period rises. The care model in this study showed that these factors had effect on long-term survivors, and on short-term trends, there were little or no changes. Thus, using GEO model to investigate miscarriage data provided more insight than the classical models. So this classical model means the normal classical Fox model, which is common, and the acceleration failure time model. Thank you.
Round of applause, please.
Hello? Hello?
Can you hear me?
Yes.
Okay, we are at a stage where if there's a question, we are going to take it, just one, if there's any burning question.
Okay.
You are a lucky one. Nobody's asking. So we are going to the next one. We are proceeding to the next one. The last presentation is going to be by Kingangi. Kingangi, are you available? We are pressed for time. Kingangi, if you are on the forum, please, we want to hear your voice.
Yes, I am available.
Can you share your slides, please, so that you present in the shortest time possible?
I don't know. Okay, how am I going to, how can I share?
There is something
at
the bottom of the screen saying share screen.
Okay.
Have you tried clicking on it, please?
Like I cannot share. Okay. Yeah.
Share screen. Share screen.
Yeah. I have clicked on it. No.
Thank you.
Okay.
Put it in the presentation mode. This one is Wordama?
It's a PDF.
Oh, it is a PDF.
Yeah.
Okay, your time begins now. Take the shortest time possible, please.
Okay. Okay, my name is Dennis Kingani, and from UOE, and I want to present my work on classification of permutations. My co-researchers are Fancy Nyapate from Chuka University and Sami Musodi, also from Chuka University. And now, this is an area, graph theory is an area in discrete mathematics. It has numerous applications. For instance, it's applied in computer science, in practical problems in combinatorics, chemistry, in chemical isomerism, also in operational research, in biology, when dealing with genetics, among other areas. As a result of these applications, it has attracted much attention to researchers in various areas of graph theory, including graph labeling, graph coloring, combinatorics, graph isomorphism, matroid, among others. Now, researchers in this area have also worked on specifically on permutation graphs, paying attention to cyclic permutation graphs, including closing numbers, isomorphism, connected permutation graphs, distances, and connectivity.
Please move to
Niches,
the
among others.
methodology. Methodology,
We
please.
have a number of people who have worked on this area, including Chartrand at Harare. We also have
Can you hear me?
1995.
Please move to the methodology. Methodology.
Graph G with
Please.
labels 1 up to N.
Bwana
which is
Kingangi.
labeled, the particles labeled 1 up to N, and which are joined. The people who have worked on this area.
Can you hear me?
And our work, we just wanted to extend studying this area by looking specifically at construction of other types of or other classes of permutation graphs. Now, and therefore, our objective was to characterize permutation graphs, specifically to develop a class of permutation on a finite set, determine the algebraic properties of these permutations, and then construct graphs from the permutations, from which we were to determine the properties of the graph that we constructed. Now, on the first objective, we were to develop a class of permutations, which we denoted by uxk power n. And here, we just came up with a mapping of on a finite set, which we proved that it is a bijection, because a permutation is actually a mapping from one finite set onto itself, such that such mapping is a bijection. So the way we did this, we first took a set of n elements or objects, that is x1 up to xn, as we have there, whereby n is a natural number, and then we fixed the identity permutation alpha naught, which consisted of the elements of n as in the order that they are shown in the set n. Therefore, the identity permutation alpha naught is the same as the set n, and then we fixed a natural number q, which defines n. So we take a natural number q, and then we partitioned q, the set n into q cells. So we have cell C1, C2 up to CP, it's supposed to be Q up to CQ, where an element xi will be in cell Cj if and only if I is equivalent to j modulo q, which means if we define I minus j by if we define the I by q, the remainder is j. And then the partition of n will allocate members of n into cells Cj for j ranging from from 1 to q in a manner that the relative positions in the set, the original set n, are preserved, so that the first element will always the first position will always be reserved, even if there is a swap in the elements x1 up to xn, then the relative positions will always be maintained. So this one led to partitioning of the set N, and this partition we denoted it with the Pn power n, and the initial partition such that an element xi will belong to cell Cj if and only if I is equivalent to j modulo q. So we fixed from here, we fixed up an element xk from set N. And then we took the union of the cells, the cells in the partition. We took the union in such a way that the cell the cell containing the element, the fixed element xk, is in a certain position t, for instance, where t is ranging from 1 up to q. Remember, we have q cells. So xk must be in one of the cells from cell C1 up to CQ. And then the union of the cell Cj, where j ranges from 1 to q, are such that the relative positions again in the cells are preserved. And we denoted this union. Remember, we started with partitioning set N into Q cells, and then we have taken a union of the cells with a certain pattern, such that the position of X, the cell containing Xk is maintained. The position is will be at position T, and we will denote the first permutation that we have come up with as alpha 1. So the permutation alpha 1 is obtained by mapping the initial permutation, which was the identical permutation alpha naught. Now we repeat this process in succession to obtain the next permutation, which is alpha 2, alpha t, alpha 3, etc. And therefore we come up with a mapping. Our bijection was actually consistent of partitioning a cell and then getting a union. That was one cycle of mapping, and it produced permutations in succession. Now we were able to, for a particular set consisting of n elements, whereby n is divisible by q, we are able to obtain a series of permutations, and which was actually finite. We found out after some times we repeated when we went on with the mapping, we were repeating ourselves, so that the number of permutations what we are able to obtain were finite. And therefore, we realized that that process was actually a mapping from the set n into n, which was actually describing a permutation, or else it was a bijection. And that was what we obtained in theorem 1. Then in the corollary is that, which is just a consequence of what we had in that theorem, is that those permutations were actually one part or a section or a subset of the set of all permutations on the set N. For instance, if we consider a category of permutations, okay, in our working, we considered a category of permutations consisting of either n equals 9 elements, or 12 elements, or 15 elements. For instance, if we take the first case, if n is equal to n elements, and we take an example of where elements are actually the natural numbers from 1 up to 9, and then fixed our element xk to be the element in the fourth position. In our case, coincidentally, the element in the fourth position is number 4. And therefore, the identity permutation, whereby every element is mapped onto itself, is the permutation alpha naught. Now, when alpha naught was mapped the first time, by first partitioning and then taking the union, whereby sorry, let me take it back. Okay, for instance, in cell 1, it will consist of all those elements which when divided by 3, because Q is equal to 3 here, the remainder is the same. For instance, when you define 4 by 3, the remainder is 1. When we define 7 by 3, the remainder is still 1. So the elements 1, 4, and 7 are in cell 1. Then in cell 2, we have those elements which, when divided by 3, the remainder is 2. For instance, 5 when divided by 3, remainder is 2. 8 divided by 3, remainder is 2. And cell 3, those elements which when divided by 3, the remainder is 0. And therefore, remember our xk is actually element 4, which is in cell 1, as you can see here. And therefore, we take the union of cells C1, C2, C3.
Bwana Kingangi, can you hear me?
Yes.
Try to summarize, please. Try to summarize. Don't go into the details, please.
Yeah, so we were able to obtain, okay, the first permutation, alpha 1, was the way we have here. Then we took another mapping, we obtained alpha 2. And from this category, if we took a mapping on alpha 2, we would repeat one of the cells earlier obtained, either alpha naught or alpha 1. And therefore, from this category, we are able to obtain only three permutations: alpha naught, alpha 1, and alpha 2. And the same case, we would get some permutations if we repeated for when n is equal to 12 or when n is equal to 15. Now, the second objective was to obtain the algebraic properties of the permutations which we obtained, and it was noted that the resulting permutations falls into two categories. There are those permutations which formed a group, and those permutations which do not form a group. And for those from those permutations forming a group, we consider they were obtained when the position when we considered an element xk which was in the middle position, and where n was actually a product of 2 primes, whereby one of okay, so that means n is equal to a product of 2 primes. So we are supposed to divide n into a prime number of cells, and in this case, this category of permutations actually formed the group, and we are able to prove that actually that class was closed under permutation multiplication, and also the associativity was there, was able to be proved. Identity was there, and every element had an universe there, which means that every class actually for that category formed a group. Now, we had an example here, for instance, when n is equal to 9 or 15 or 21, when n is equal to 9, we were able to get the properties, algebraic properties of isomorphism, and this is the group table that we came up with for that particular class, and we realized that that group is isomorphic to the group Z2 under addition. Also, if we did the same for the other class of 15, n is equal to 15, then that one was also, the group was isomorphic to Z6 and addition. And for the class of consisting of 21 elements, the group was isomorphic to Z4 and addition. We were able to get other properties, graph properties, from the obtained graphs, which consisted of, we checked the other properties like we have connectivity measures. We also have, we checked on those groups which graphs which were the cycles in the graph, among other graph properties.
Take us down to your conclusions, please.
Yeah, okay, for the conclusion, this research developed a class of permutation graph, which we denoted by U. That symbol is not there, but we denoted it with the UXK power n. And a variety of graph properties was gotten from that class of permutations. Now, this class of permutations, graphs can be investigated for further properties and also to see its applicability. Now, being a few mathematicians, we need someone to come in who can relate this to the real life problems. That's the end of my presentation. Thank you.
Thank you, Bwana Kingangi. Let's give him a round of applause. This is mathematical. I don't know whether somebody followed the permutations. Bwana Kingangi, you gave in-depth presentations on permutations. Anybody with a question for the presentation? There's none. So we are just left with one that came recently, and then we'll be done. Bwana Adan is all the way in the coast. Are you there? Can you hear us? Adan?
Yes, I can hear you.
Please give us the gist of your work, the major components. People will leave you and you'll be alone in the room if you take too long.
Okay.
Bring your slides on board, please.
I think Mr. Dennis Kingang, okay, was still sharing, so I cannot.
Bwana Kingangi, and the sharing. Bwana Kingangi. Thank you. Are you able to share now?
Yes, can you see it clearly from there?
Yes, we can see it. Put it in the presentation mode, slideshow.
Okay.
We welcome you to give us a presentation in the shortest time possible, and your time begins now. Thank you.
Okay, good afternoon, everyone. My name is Mr. Adan Jilo. I'm a student at Technical University of Mombasa doing an MSc in chemistry, specialization in analytical chemistry. My work involves synthesis of nanoparticles, silver nanoparticles, which later I'm going to use for degradation of organohalide water contaminants commonly found in rivers and oceans. So my title of the project is Silver Nanoparticles for Reductive Dehalogenation of Emerging Organohalide or Aquatic Pollutants. My supervisors are Dr. Ali Shea and Dr. Alois Ogweno. So for the background of information, I'll just brush through. The emerging halogenated contaminants, they can enter the ecosystem and they are usually resistant to degradation, both mechanical and chemical. So the substances can accumulate in higher concentration as they move up the food chain, posing risks to both wildlife and humans who consume contaminated organisms. So the most common
You can move on, please.
in Kenya.
Move on, please.
Okay.
Thank you.
Okay, then I'll go to the main objective is to evaluate the potential of silver nanoparticles in removal of emerging halogenated water contaminants. My specific objectives are to prepare and characterize the silver nanoparticles, to establish the halogenation patterns of selected halogenated emerging contaminants by use of the silver nanoparticles in water, and to evaluate the stability and recyclability of the silver nanoparticles for the dehalogenation of selected halogenated emerging contaminants in water. So I used three methods for the preparation of the nanoparticles. The first one for sodium borohydride was used to check to determine the correct concentration of the silver nitrate. So the first one was reduction of silver nitrate with sodium borohydride. The second one was reduction of silver nitrate with ethylene glycol. And the third method is green synthesis, was reduction of silver nitrate with aloe vera extract. So the following are the procedures that I followed for preparation, the reduction of sodium borohydride. So here you put the borohydride on the burette and the sodium and the silver nitrate was on the burette and the sodium borohydride was on the beaker. So it was open dropwise while continuously stirring. So when the dropwise addition was completed, the color changed from clear to yellow. That was an indication of the formation of silver nanoparticles in water. Then that mixture formed was taken for analysis in UV spectrometer. The second method was by ethylene glycol. So several concentration of ethylene glycol were prepared, also mixed with silver nitrate, and they were placed on a water bath as shown there. And it was heated at 80 degrees for 3 hours. After the 3 hours also, the clear color changed to yellow, indicating the formation of silver nanoparticles. And for the green synthesis, here I used the aloe vera extract, which is the flesh, the white flesh. Also, it was extracted and several concentration meds, and they were by volume. They were added and let to stand for 2 hours. So here there were two methods. One was let to stand at room temperature, and another one was heated. This is according to already done work. So I was trying to look which one gave a higher absorbance, the formation of the nanoparticles. Also, the color was coming to yellow. So this is the equipment used for characterization. It's a UV visible spectrometer found at Mombasa Government Chemist. So this is from Suman et al. Showing projecting the formation if nanoparticles are available and you run it through UV spectrometer, you're going to form such a spectrum, absorbing at around 400 to 450. So for the sodium borohydride and silver, I tried to vary the concentration of borohydride and also vary the while keeping the other constant to try to look for the best, the optimal concentration which could lead to formation of more nanoparticles, which I'm going to use next now for the degradation. So from this, it was showing decreasing lambda max with increasing sodium borohydride concentration, implying decreasing nanoparticle sizes. So from the photos of the beaker there, you can see the lower the concentration, the lighter the color of the solution. Then when you vary the silver now, it was showing that higher concentration of silver nitrate was leading to agglomeration by formation of the dark colors of beaker 4 and 5. So this is just an illustration to show the effect of the silver nitrate. So for ethylene glycol also, the variation, the best combination was found at around 20%, giving the highest lambda max and highest absorbance. For aloe vera, The other lower concentration of aloe vera was giving absorbance which are much higher and are not in the range of the nanoparticles. So I ignored them in the reporting the results. So I concentrated on the 3. And from the results, there was no significant difference between when you heat and when you don't heat. The better if you don't heat, because somehow the lambda max was high and also the absorbance compared to heating. So as when the nanoparticles were formed, with the 3 methods, I stored them in the dark, was due to silver nitrate usually breaks down, agglomerates due to UV light. So I'm trying to check now the stability of the formed silver nanoparticles. So the first one I checked on 1st of February, and the second one I checked at the end of April, and the stability, the absorbance was still found to be within limit plus the wavelength, showing no agglomeration or gradation. So for the conclusion, said that silver nanoparticles were formed by reduction of silver nitrate with sodium borohydride, xylene glycol, and aloe vera extract. So this reduction was caused by the hydride, which is the hydrogen part of the sodium borohydride, xylene glycol, and phytochemicals in aloe vera. This was indicated by absorbance in UV visible between the range of 400 to 430 nanometer, characteristic of silver nanoparticles. That was for that. So these other ones are still ongoing. The phytochemical characterization of aloe vera, this is to determine now the exact part of the aloe vera which causes the reduction of the silver nitrate, and the antibacterial characterization of silver nanoparticles, reductive degradation of organohalide bromobenzene. This one has I still have some challenges due to breakdown of the GCMS, but the work is still ongoing, and the recyclability test will be done after the degradation, the organohalide, example bromobenzene and others. So I'd like to acknowledge the Technical University of Mombasa, supervisors Dr. Ali Shea and Dr. Ogweno, government chemist, Mombasa branch, and University of Eldoret for giving me this chance to present my work. Thank you.
Let's give a round of applause to him. Somebody with a question? This work was to do with water quality. Am I right?
No, yes.
So the samples were collected from rivers and the ocean.
Yes, but I've not reached that stage now. That is the degradation stage. That I have to use the GCMS to see the breakdown of those, yes.
You have escaped my question, so let's give you another round of applause. We are coming to the end of this session. I'm requesting everybody to move forward so that we can formally close the presentations. I want to thank all the presenters, especially those who have done their presentations virtually. I also thank the other team from the other room. We are coming to the close of the conference, and I want us to be as close as possible. Please, if you are at the end, try to move close. Without any further ado, I want to thank each one of you and hand over everything to the directorate. The professor, please, thank you for the session. Take it over.
Yeah, thank you so much, participants. I would like to thank the management for supporting this conference, as they always do. Our division, Directorate of Planning, Research and Extension. I want to thank all the participants, the keynote speakers that came and gave us very enlightened. Actually, they really enlightened us. I'm giving this vote of thanks because DVC ASA is going to close, and once she closes, we cannot open again. So I'm giving my vote of thanks now. There's a team that was working behind the scenes, the committee, the catering, central services, estates, all those team that you don't see here, they've been working behind the scenes to make this conference a success. So I thank all those people. There is that other team there. You can see the complicated gadgets that we have there, the ICT, the photography team. Thank you very much for ensuring that everything went according to plan. So with that, I want to call on DVC, Academic and Students Affairs, to close this conference. I hope we are not going to open it again. Once it's closed, it's until another time again when we have another conference. So DVC, kindly come and close, and then we shall have someone pray for the lunch after that.
Colleagues, ladies and gentlemen, who have been very supportive participants of this conference, good afternoon. I am glad that we have sat through this meeting up to this moment, and that shows your commitment on making sure that you network with other people and get to know what they are doing, and also share with them what you are doing. Having a scientific conference is a great learning platform for all of us. And I must say that all of us have something to learn. We are each at different levels in our careers. And we should all retain the perception that everybody has a chance to learn something new. As I stand before you this afternoon, sitting through these sessions has been a great opportunity for me to learn. And one of the things that I take home with me today is packaging. When you have a gift packaged and placed somewhere, you are not sure what is inside the package until it is unwrapped, and then we get to see the contents. So this conference has been an opportunity for us to have several gift packages to society unwrapped for us. And I do appreciate the effort that each one of you has made to ensure that you open your gift to the rest of us so that we have a share of it. Thank you so much for the work that you have done. Thank you for the papers that you have presented. Thank you for the knowledge that you have shared. Keep making good packages and opening them to the world to see and to have a share of the gift and the knowledge that you are endowed with. I want to thank the directorate and the division that organizes these sessions for us. Yesterday we were challenged that the problem we have as scientists is that we sit in our little corner and talk to ourselves. I want to believe that if we take that as a positive advice, in future, we should be able to interact on a forum like this with people from other specializations, for example, the communicators, the language experts, so that we are able to learn more even on how to package ourselves. Because at the end of the day, it's important that the audience gets to know what you are talking about, gets to know what you are doing, and becomes part of your processes. As scholars, and some in their early careers, I want to encourage them to keep this practice, because you will get to learn a lot from interacting with those who have had more experience in your specific fields. As the presentations were going on, I am sure you have identified some people who have similar interests as you. This should not be the end of the interaction, but instead I encourage you to reach out. Through the directorate, I'm sure you can get their contacts and be able to talk to them and build your networks. In science, we rely a lot on building networks. Not just in science, but in academia, we rely a lot on building networks. So I encourage that you follow up on those you have identified as potential partners, so that in future, if you wish to write a research grant, you will know whom to talk to and who to collaborate with. The university management is in full support of our progress in research and innovation, and I would like you to remain a part of that process, so that we move from where we are to the next level. I believe that every opportunity that comes for us is for us to make an improvement. We may have had 50 presentations in this conference, but in future we could do more and improve our contributions, and there will be more opportunities like this coming to you. I am assuring you on that on behalf of the management, because in our budgeting, we always consider that research is one of our key mandates as a university. So let us continue learning from each other, and let us continue doing what we do best and reaching out. With those remarks, I would like to officially declare this conference closed.
So one more time, let us humble ourselves before God as we pray and thank God for being with us. Let us pray. Father, in the name of Jesus, we give you honor this afternoon, Jehovah, because you have been with us. We thank you for the success. We thank you for the smooth flow. We thank you, God, for grace for each one of us who have been able to be here and to present. And we pray, Jehovah, that you continue to walk with us even as we go through mentorship and do research together with the university. Lord, as we break for lunch, we thank you for the meal, and we pray it is sanctified. As we go home, we pray for safe journey mercies, for it is in Jesus' name we pray and believe. Amen.